Just waiting for the nod. I'd like to acknowledge the traditional owners of the lands on which we meet today, the Wurundjeri and Bunurong peoples of the Kulin Nation, and pay my respects to their elders past, present and emerging, and acknowledge also the traditional owners of the lands on which people are joining us virtually. Special welcome to the family of Professor Derek Denton in attendance. His sons, Angus and Matthew, and their families, Paulina, Annie, Olympia and Josh. Welcome to ladies and gentlemen and distinguished guests. It's my great pleasure to be hosting today's Festschrift in honour of the late, great Emeritus Professor Derek Denton, hosted by the Florey Institute. We all knew Dick as a pioneer in medical research and in scientific leadership. Where we are today, he had a clear vision for a future where curiosity-driven research was valued and improved the health of everyone. As a scientist, he led international collaborations on instinctive behaviour and was elected to the most prestigious science academies in the world. And as a peer, he was a brilliant listener, and he went to great lengths to enrich people's lives and provide opportunities for them. I'm Robbie Williams, and when I first met Dick and his wife Maggie 5 decades ago, I was amazed by the extent of our common interests in enology, the deep study of wine with its practical applications, science as a broad investigation, ballet, art, they knew what I liked, ideas, more enology, and also a suspicion of ideologues and deniers. However, science was the true love. And so today we have the opportunity to learn from a series of fascinating scientific presentations from some of Dick's closest collaborators and friends. We'll hear speeches from leaders of the University of Melbourne, the Florey, and from his son Angus. It's then my pleasure to deliver the Derek Denton oration in science and the arts. And finally, to finish off, we'll have lunch. And as we all know, Dick would have wanted a toast to his memory. Housekeeping, right. Our event photographer will be snapping away. If you do not want to feature in these photographs, please speak to Samantha Sims. Sam, stand. Tell her you don't want it during the breaks, and we'll make sure you're not featured in the final photography. This is a live streamed event. We have several cameras set up for those viewing virtually, and this will be available afterwards. Toilets are located on my left, your right, up the stairs, ramp next to the DAX Centre, and can be accessed via the door at the back of the auditorium, and through the door to my right, your left, that one. We'll punctuate this event with regular breaks and morning tea. That'll be at 11 a.m. and lunches at 1:30. First introduction of today's event is Professor Jim McCluskey, Deputy Vice-Chancellor of the University of Melbourne, to share a few words. Please.
Thank you, Robin, and good morning, everyone. And let me just acknowledge Dick's family and also some of the former directors of the Florey I can see in the room, and of course Trevor, the existing director. Well, a picture is worth 1,000 words, and there are quite a few images I have of Dick Denton that capture his personality and aspects of his life. There's a portrait hanging on the 4th floor of the Florey, and another smaller portrait that I enjoy looking at whenever I visit the Royal Society in Carlton House Gardens in Waterloo, London. Beautiful pictures that depict a man of considerable intensity, with laser-like eyes and a confident smile. And he has lots to smile about. He's had a wonderful life, a wonderful scientific and personal life, quite remarkable. And there aren't too many Australians of such eminence that they have a portrait hanging in the company of Huxley, Darwin, Hooke, Newton. They're iconic names in science. It speaks to a level of distinction that is pretty rare. And we celebrate that today as his scholarly community at the University of Melbourne and the Florey Institute, following from yesterday's state service. The most explicit portrait of Dick the scientist is in the National Portrait Gallery in Canberra, and there's a copy of it here by Everett Pluke. And you can see this is called The Equation of Life, a portrait of Professor Dick Denton. It has Dick behind a sheet of perspex, and there are notes and formulae and illustrations and arrows relating to the rich tapestry of his scientific work visible to us as he half grins but still has the demeanour of a pretty serious scientist. For me, this portrait conjures that relentlessly active mind. Certainly that's the man I knew, and I didn't know him as well as many in this room, but I think of him as a quintessential polymath. The sort of scientist facing extinction in the modern funding environment. He was a scientist who knew a great deal about a vast number of different subjects and virtually everything about his own subject. If he was wrong, I wouldn't have known it. Among his authored books are several that explore the biochemical and neurological basis of self-awareness and consciousness. The Hunger for Salt, which I spent the last week browsing, and it is an astonishing tome. And then The Primordial Emotions. So this reflects a quality that Dick had. He was able and confident and ambitious enough to ask really big, hairy, audacious questions that actually, frankly, scare off many other scientists. It speaks to a particular kind of mind that is not intimidated by the vastness of the intellectual landscape that he's about to traverse. He stood for an unrestrained style of scholarship and curiosity about the whole world. To me, more reminiscent of Thomas Henry Huxley or Charles Darwin, philosopher-scientists who didn't see the difference between art, science, philosophy, etc. Another portrait reveals yet another side to this man, and I've only just been made aware of this. It's at Trinity College, where Dick was awarded Lifetime Fellowship, the highest honour that the college can bestow. But there's a bit of a backstory. Dick, many of you will know, was, in the English language term, sent down from Trinity, expelled. Expelled because it turns out he was manufacturing sly grog. And this sly grog, manufactured on the grounds of the college, was the substrate for many boisterous and noisy parties, which were the cause of his ultimate expulsion. Pretty plucky entrepreneurial initiative, pretty consistent with the man that I knew, actually. But it didn't stop there. He was a boy from Launceston. He had no housing in Melbourne, and he'd been kicked out of his only home. So his friends ensured his survival by sequestering him away secretly in the loft of the Bean building, which is the long building on Royal Parade, unbeknown to the warden of the college. And I don't know how long he lived there, but I'm led to believe that he continued his experiments in applied chemistry. Now, I think that portrait reveals two sort of interesting aspects of the man. When he was invited, decades after that, incident at Trinity, to become a fellow of the college, quite understandably, he was a little bit testy about that, and perhaps not immediately impressed with the belated recognition of his brilliance. But then he consented and put the question to them straight away, and what about the portrait? And that's the origin of the portrait. So this cheekiness was part of his makeup. I think it remained part of him well into his 90s. But it also reveals a man untroubled by self-doubt, untroubled by his self-image. He just had the balls to ask. And then the second thing I think that's interesting about that portrait, and I don't have a picture, but it shows him in a blue shirt with blue slacks, relaxing, and there's a table, and on the table there's a glass of red wine. And he has a slightly pained look on his face, which I read as a gentle message to Trinity, his alma mater, and the history of that time in that college. Dick was fundamentally a physiologist. Modern physiology, I think, struggles to exist in its own right in the way it did when I was a medical student. It's been sort of co-opted, eroded, stolen, pilfered by other disciplines, genetics, biochemistry. These disciplines tend to be far more focused, biochemistry generally about individual chemical reactions or sets of reactions, but systems, which are what physiologists study, whole systems, complex, interconnected, modulating influences, far more complex. And the questions that you ask lead to far more complex answers. The regulation of complex things like blood pressure, blood glucose, temperature. These are challenging whole systems. And I think, again, a really interesting reflection of Dick Denton, the man. He wanted to understand the whole canvas, not this corner. And that stands in contradistinction to a lot of modern-day science and research that is driven by wanting to solve individual, small, focused problems. I also have a social image of Dick Denton. It comes from a photograph rather than a painting. It's one of my favourites. Many of you will know it. It's a picture of mostly men in tuxedos, standing around, obviously relishing each other's company. Black and white photograph. Dick was seated, turning towards one of the people in the picture, Dame Hilda Stevenson, who endowed the Stevenson Chair of Paediatrics at the university, with a glass of wine in his hand, smiling and saying something undoubtedly enormously witty. And in that group there was Kenneth Meyer, Ian Potter, Pansy Wright, Nugget Coombs, effectively the people who held the levers of Melbourne, who ran Melbourne. And he was just so comfortable. He was just so at ease in that company. It was taken at pre-dinner drinks after a board meeting of the Howard Florey Institute in 1971. It's a historical record, not a hysterical record, a historical record. It's a very elegant photograph, and there's no question it tells a huge story. And as many of you know, it was Kenneth Meyer and Ian Potter who became critical to the creation of the Florey, and not in the picture, just to emphasise the company that Dick kept, was Sir Evelyn de Rothschild, the personal banker to Queen Elizabeth II, who was on the board of the Florey. So Dick was really comfortable operating in these different environments, but these were authentic friendships. Yes, you know, we all know what he was chasing, but he did it with such charm and style and authenticity. And it's an important part of his legacy. I think it's quite a challenge to be able to interest people from other worlds in science, technology, the human mind, physiology, in the way that Dick did. It was really important to the enormous influence that he wielded. It was really important to the creation of the Florey. So he is one of the great figures of the University of Melbourne. He created the Florey itself, a great and continuing legacy. He did it with the support of the Meyer family, Ken and Bales, and more recently Martin, Sir Ian Potter, Carrie Packer, the Kleberg family, the Rockefellers, Evelyn de Rothschild, all the people I've mentioned. His impact on generations of students, colleagues, people he's mentored, influenced, has been enormous. He is a key member of our community right up to the last year, even in his 90s. He endowed the Derek Denton Oration in Art and Science at the University of Melbourne, the inaugural oration given by Lord Martin Rees, who I think was Astronomer General and President of the Royal Society, Robin, yes? So what a legacy, and I think a legacy that reflects a conscious effort to encourage the arts and sciences to come together. I suspect, not unrelated to his remarkable marriage to Dame Margaret Scott, Maggie Scott, ballerina and one of the founders of the Australian Ballet and the Australian Ballet School. A kind of marriage of the arts and sciences, although I suspect there might have just been a little bit of grit in there as well. His work in the broader community should not be overlooked. I would say that between Dick Denton and John Funder, all the bad salt in foods that we eat was progressively removed. In Dick's case, it was Heinz baby foods, convinced the company to desalt them. And I know that John Funder took the salt out of Vegemite. You wouldn't know that actually if you had some. So they're a remarkably interesting pair of scientists. I actually think of them a little bit in the same part of my brain. They're sweeping intellectuals, polymaths, people with a large vision. And, you know, Dick, being the big figure he was still at times a prophet, save in his own hometown. He was just a little bit insecure at times, not very often, but he gave a lecture at the Collège de France in Paris, and he reported that he was to talk on his theory of consciousness. But the college was very poor at advertising these things, and he was told privately he'd expect about 10 people. And he got there, and of course, it was overflowing. The French, having that great respect for Dick's exceptional scientific mind, an intellectual celebrity 12,000 miles away. Dick's exceptional scientific mind was recognised by just about all the important scientific learned societies, the Royal Society of Physicians, the French Academy of Sciences, the American Academy of Arts and Sciences, the Australian Academy of Science, foreign member of the Royal Swedish. He was also a judge for the Albert Lasker Award. And in a sense, all of these prestigious forms of recognition spoke to his brilliance. I had one encounter on a personal level with Dick. Out of the blue came an invitation for dinner. Dick Denton, myself, and the then Dean of Medicine, Kshitij Kapoor. And I said to Kshitij, "What's all this about?" He said, "I don't know." We got there, and there was Dick reclining in a plush Chesterfield leather wingback chair with a glass of red wine. And it was just a warm, wonderful evening. I suspect the agenda was to tell us how to run the university. But he got so sidetracked with his sort of inveterate name dropping that it became a complex history lesson with a bit of you need to do this and you need to do that. But you know what? It was just such a wonderful privilege, and the quality of the wine was outstanding. Towards the end of his life, Dick became very anxious about his library, a kind of symbolic surrogate of his legacy, I would guess. He made a big fuss about it, and it was Mark Hargraves and the archivist in the Faculty of Medicine who, with a bit of help from Trevor, put this in boxes, labelled it, stored it properly, and made Dick very happy. It was a fantastic gesture. Some of these volumes are in the bookcases in the Florey. Authors include Koestler and Dennis William Brogan's The Development of Modern France, 1870 to 1839. I haven't read that one yet myself. Derek was a rare individual, a mix of engaging personality, charisma, you might call it Hawke-esque charisma, charm, intellect, and confidence, each component as bright as the other. So I say personally, Vale, friend and colleague, we celebrate his scientific excellence today in today's Festschrift, and I'd like to invite Trevor Kilpatrick to the podium. Thank you.
Thanks very much, Jim, for that great address. And to summarise, I think what we're recognising is the power of an amazing individual who has a holistic vision. who sometimes is working in the context of bureaucracies which have a more myopic vision. And that represents a challenge for us even now in 2023 as to how we nurture rare individuals like Dick, the real polymath as you indicate. And I think that's something we should reflect upon during the day. So what a great address, and very much appreciated, Jim. Now, I'd also like to turn to Robin and thank Robin on behalf of us all for everything that he's been doing for us over the last 2 days. Perhaps not many of you know that Robin has just flown in from overseas. He's still jet-lagged. His luggage was lost, his credit card was lost, he's borrowed a suit. I won't ask which carrier it was, Robin, but we're sympathetic and we're very appreciative of what you're doing for us as well. I'd also like to add my acknowledgement to the traditional custodians of the lands on which we meet, the Wurundjeri people of the Kulin Nation, and to pay my respects to their elders past, present and emerging. I'd also like to pay my deepest thanks and gratitude to the Denton family who are here to celebrate Dick's life with us today, and in particular to recognise their ongoing contributions to us at the Florey. So thank you. I'd also like to acknowledge the organising committee and those assisting in bringing the day together, in particular Samantha Sims, Georgia Brown, Ryan George, Lorraine Tadd, Liz Brentnell, Kate Anderson, John Roberts, Anthos and Sarah Hancock. So now let's turn to the scientific presentations. We all very well know that Derek Denton also liked to be known as Dick, and he was committed to the furthering our understanding of instinctive behaviour, including the response to thirst and hunger, and how we maintain homeostasis through both cerebral and neuroendocrine mechanisms. Dick's commitment to this field of research has had tremendous impact. I don't think it's hyperbole to state that many lives have been saved and many important research projects and careers have been built on the basis of his research. He was truly a trailblazer. Dick was passionate about collaborating, hearing and learning others' scientific views, and nurturing the next generation of influential scientists, as we'll hear today. So it's only appropriate that in today's Festschrift we hear from a variety of Dick's key collaborators, his protégés, and ultimately his friends about the impact he had on their science, their experiences in working with him, and ultimately his impact upon their lives. Now, the first of the scientific presentations is from Dr. Philip Ryan on the neural circuitry that controls fluid and salt intake. Phil has come a long way since completing his PhD with us in 2013, and he's since been awarded at least 2 prestigious awards to continue his overseas studies, including an NHMRC C.J. Martin Fellowship. And after spending 4 years at the University of Washington, Seattle, he has returned to work at the Florey as a postdoctoral fellow, focusing on behaviour neuroscience-oriented research using cutting-edge tools and techniques to trace and characterise neural circuits. So without further ado, Phil Ryan, thank you very much.
So thanks very much, Trevor. And I really appreciate the opportunity to be able to present my work here today. It's been a really great honour and privilege getting to know Dick, particularly over the past 5 to 6 years, where I've been involved in a collaboration with him and several others here as well today. So today I'll present my research looking at the neural circuitry of fluid and salt intake. And quite a bit of this work was done at my time in Seattle, where I did my postdoc, and then continued on here. And a lot of this work overlapped with a lot of the work that Dick and others had done at the Florey for the past several decades, which is one of the reasons why I became involved in a collaboration. And one of the things I really enjoyed about working in this collaborative capacity with Dick was his constant enthusiasm and passion for science. So as I said, I've been collaborating with Dick for about the last 5 to 6 years. And one of the really enjoyable things about collaborating with Dick was not just the conversation, and there were some great scientific conversations, but often the venues in which we held these conversations. So there was dinner often at University House or at his place or at other venues, and there was dinner often accompanied by some wine, which helped get the conversation flowing. And I think I learnt from Dick the importance of these informal conversations to help with science. I think it helped us discuss ideas and also come up with some new and fresh ideas as well. So on the right of this slide, I've put two of Dick's books. The Hunger for Salt has already been mentioned, so that's one of Dick's main works, looking at salt and salt appetite. And in the bottom right, I've also put one of his books, The Primordial Emotions, which has also been mentioned. And primordial emotions are those subjective feelings underlying various instincts like hunger, thirst, and salt appetite. And I think Dick was particularly interested in studying these because he thought that they'd provide a window into understanding consciousness, which also helped with some of the conversations at dinner. And they also provided an influence for some of our collaboration, which I'll get on towards at the latter part of this talk. So just before I go into my work on the neural circuitry of fluid and salt intake, just thought I'll briefly mention, during my PhD, Dick was editor on one of my main articles, which was looking at the neuropeptide relaxin-3 and its receptor RXFP3 and how they regulated alcohol seeking. And in this particular paper, I found that if I injected the antagonist to the receptor, that it decreased alcohol drinking or self-administration and also decreased relapse to alcohol or reinstatement. And while I'm not going to talk too much about relaxin-3 today, relaxin-3 was also involved in increasing food and fluid intake. And when I was looking around for a postdoc, I was really interested in understanding a bit more about the neural circuitry underlying food and fluid intake in more detail. And in particular, looking at the brain circuitry underlying satiation signals. So just really simplistically, if we're hungry or thirsty, we might eat or drink something, and often to the point of satiation. And if we go past that point, it can feel quite uncomfortable. And then for a few hours after that, we might feel satiated, and then we might eat or drink some more. Now, these central signals, so brain signals, are really critical for energy and fluid homeostasis, along with a lot of the peripheral signals as well. And if they're energy homeostasis, I suppose homeostasis is the balance within the body. If it's dysregulated, it can lead to disorders like obesity, and often fluid homeostasis can be dysregulated in disorders like heart failure, kidney failure, and liver failure. So I was really interested in looking at the brain circuits or the neural circuits underlying satiation, in part because I thought it'd be interesting to understand how they work in the brain, but also potentially to develop some targets or to identify some targets for potential therapies. And so I was particularly interested in using some of the new mouse genetic techniques which had come out over the past decade or so, which have really revolutionised neuroscience. So a lot of these genetic tools and techniques have really allowed us to selectively target and study the function of and the neural circuit maps of specific groups of cells within the brain. And one of the key centres for this was in Seattle at the University of Washington. So during my postdoc, I went to Seattle, which is in the Pacific Northwest of the States. And here's a nice image of Seattle with a Space Needle and Mount Rainier in the distance. It was a very picturesque city. And there's an image of the University of Washington, which is where I performed my research. This was the library and another area. The lab wasn't quite so picturesque, but it was a good lab. And there was my supervisor, Richard Palmer, who's a Howard Hughes Medical Fellow, and also had been instrumental in developing a lot of the genetic techniques which I was using. And so while I was doing my postdoc, I identified or discovered a group of neurons which are involved in controlling fluid satiation. And I thought today, as part of this talk, I'd just take you through, sort of briefly, an overview of how I investigated and characterised this group of neurons that controlled fluid satiation, before I talk a bit about the collaboration I did with Dick. So just as an overview, to investigate a neural circuit looking at using some of these genetic techniques, it's firstly important to select a brain region and expression markers, and then to selectively target these brain neurons using techniques like Cree mice, which I will talk about. Then it's important to investigate the function. So I did this by artificially activating selected neurons using techniques like DREAD receptors, which I'll talk about. And as well as activating these artificially, it's also important to check their function under normal physiological circumstances. And for this, there's some techniques like FOS, which is a marker of neural activity, and also calcium imaging, which allows us to look at the neural activity in real time. And then I could trace out the neural circuit map as well, to sort of help understand the neural circuit that was underlying all of this. So I'll just take you briefly through all of these in turn. So firstly, selecting a brain region and expression marker to look at satiation. So just prior to going over to my postdoc in Seattle, there'd been a paper that had come out suggesting that oxytocin might play a key role in regulating food intake. And there'd been several other papers before that which had also shown that, but it looked like it could be a key target to look at for exploring this function. And also other papers had suggested that oxytocin might play a role in fluid intake as well, which is one of the reasons why I was interested in investigating this system. But oxytocin is probably better known as a hormone and neuropeptide that is involved in inducing labour and lactation and social behaviours, and also potentially in decreasing anxiety and addictive-like behaviours as well, which is one of the reasons why I thought this was a really interesting system to study. And the brain region that I chose was the parabrachial nucleus, which is located in the hindbrain. And here's just a mouse sagittal section, and the PBN is here. And this is known to play a key role in satiation behaviours. And in Richard Parmiter's lab where I was, the parabrachial nucleus had been well studied for its role in food satiation. And I just checked to see whether there was oxytocin, or the oxytocin system was involved here, and there was some oxytocin receptor expression in quite a few of the neurons in the parabrachial nucleus. So here you can see an image with its expression in red in two of the subregions, the dorsolateral and the external lateral region. So it seemed to be a good region to, I suppose, study to look at its function. So then needed to know how to selectively target these brain neurons. And for this, I used a Cree mouse. So Richard Parmiter was well known for developing these types of mouse lines, which allow us to selectively target groups of neurons. And he developed a mouse line called the oxytocin receptor Cree mouse line, where he inserted the Cree recombinase enzyme downstream of the termination codon of the oxytocin receptor. So that meant that any neuron or any cell that expressed oxytocin receptor would also express this Cree recombinase enzyme, which allowed us to selectively target these neurons. And I'll just give you an example. So if I injected, pardon me, a virus, an adeno-associated virus, or AAV, with the fluorescent tag mCherry, and here you can see it's in inverted format into this brain region, it would selectively invert and express or fluoresce red in the neurons, in the Cree expressing neurons, so in the oxytocin receptor expressing neurons here. So it's a really nice way of selectively targeting these oxytocin receptor expressing neurons in the parabrachial nucleus. And you can see an image of that on the right-hand side. And I'll call these oxytocin receptor PBN neurons from here on in. Okay, so now that I can selectively target these brain regions, I was interested in activating them to explore the function. And for this I used DREAD receptors. So DREADs are designer receptors exclusively activated by designer drugs. And the stimulatory DREAD, HM3DQ, is a modified human muscarinic receptor. So typically muscarinic receptors respond to acetylcholine to enhance neuronal excitation. These have been modified so they no longer respond to acetylcholine, but rather to the designer drug CNO, or clozapine N-oxide. And when you inject CNO intraperitoneally, it'll enhance the neuronal excitation, and the effect tends to last for about 4 to 6 hours. So firstly, I want to see whether activating these neurons might decrease food intake. And so I injected the stimulatory DREAD along with a fluorescent tag mCherry so that all the neurons would fluoresce red into the parabrachial nucleus. I left it for about 3 to 4 weeks for the virus to express. And then prior to doing behavioural experiments, injected CNO and looked at both baseline food intake, so mice tend to eat most during the dark cycle, so at the start of the dark cycle, and then after refeeding after a 24-hour fast. And here I found that activating these neurons didn't alter food intake, so there was no significant difference at baseline or after a 24-hour fast. And here in red are the HM3DQ injected animals, or the DREAD injected animals, and in blue are the mCherry control animals, so they'd just been injected with the fluorescent tag, so they were a good control. So given I hadn't seen anything with food intake, I also looked at water and salt intake. And for this I used a 2-bottle choice paradigm. So mice had access to water and 0.3 molar of sodium chloride to assess the volume and osmolarity. And these mice were on a salt-deficient diet, so that all of their sodium chloride came through the sodium chloride solution, so it allowed us to accurately measure that. And so I looked at baseline fluid intake, fluid intake after dehydration, and also after salt depletion, using the same paradigm as before with the food intake. And so, interestingly, at baseline, so at the start of the dark cycle, in both tubes there was a significant decrease in sodium chloride and water intake, suggesting that it has an effect on fluid intake. Then I looked at saline and water intake after dehydration. So here the mice were habituated to the saline and water for several days, then I removed the fluid and returned the fluids after injecting CNO. And here there was really quite a substantial decrease in both bottles, so both the sodium chloride and the water intake, and in total the decrease was about 80%. So it suggested that activating these neurons had a really substantial effect on fluid intake. And I was also interested in looking at salt appetite as well. And to induce a salt appetite in mice, I once again habituated the mice to this 2-bottle choice, and the mice were on a salt-deficient diet. And then injected furosemide, which is a salt-depleting diuretic, and then removed the saline. And then after 48 hours, returned the saline to check. And here I found that there was no significant difference in the saline intake, but there was a substantial decrease in water intake. So it suggested that activating these neurons decreased fluid intake at baseline and after 24 hours of dehydration, but didn't decrease food intake or salt appetite, suggesting that they were selective for decreasing fluid intake. So given I'd seen this effect on fluid but not food intake, I also looked at a liquid diet, and in this case used Ensure, which is a highly caloric liquid diet, often given to cancer patients to help them maintain their weight. And mice love it. But activating those neurons didn't decrease this liquid diet, and the water intake here was low, so there was no significant difference here as well. So I suggested that activating those neurons was selective for decreasing non-caloric fluid intake. And so I'd artificially activated these neurons. Next, I wanted to just assess what their activity was under normal physiological circumstances. Are they involved in fluid satiation type behaviours or fluid regulation behaviours? And so for this, I used an oxytocin receptor Cree mouse, which had been crossbred with a reporter mouse line, so that all the TD tomato, so that all the cells expressed red. And then, so I looked at fluid intake, removed fluid, and then returned it 24 hours later, and then perfused the animals. And in a second group, they remained dehydrated. And I looked at FOS expression, and I think I mentioned before that FOS is a marker of neuronal activity or activation. And in the left-hand side are the red, so the oxytocin receptor expressing neurons. In the middle are the FOS expressing neurons showing neuronal activity, and on the right is the merger. And as you can see, there was increased FOS in the fluid returned animals, but not in the fluid depleted animals, and about 20 to 30%. So this was a good suggestion that these neurons were involved in the rehydration and satiation responses. Also injected a genetically encoded calcium indicator, GCAMP6, and this, genetically encoded calcium indicators fluoresce when there's increased neuronal activity in the neurons. And then I could implant a miniature microscope so you could actually look at the neurons as a mouse was behaving in real time. And here's just a picture of a mouse with a miniature microscope on its head. And I'll just quickly play a video. If I can. Do you mind just quickly coming up? It's just not going down to that bottom left. There we go. Thank you. Great. And so that mouse has been dehydrated overnight, and in the bottom is the fluorescence from the parabrachial nucleus. And then when it starts drinking, you can start to see fluorescence in a lot of the neurons in the parabrachial region. And as it drinks more and more, there's increasing fluorescence. So if we look at some graphs or some raster plots, at the 0 time point is when the mice start to drink. And you can see about 50% responded to water, whereas few responded to Ensure or an empty bottle. And so I suggested that there was increased calcium activity in these oxytocin receptor PBN neurons after fluid return, but not Ensure or an empty bottle. suggesting that these neurons were activated by fluid satiation. And so I've looked at the function of these neurons, and next I want to trace out a bit of the neural circuitry as well. And so I was looking at the downstream projections of these oxytocin receptor PBN neurons, and for this I injected synaptophysin, which is a tracer, which will track down to the terminations so you can map out the circuitry of the first order projections. And I found that there was some major downstream projections, particularly to the some fluid-related brain regions. So the OVLT, or organum vasculosum of the lamina terminalis, and the median preoptic, or MNPO, both of which have been studied quite a lot here at the Florey, and also the central nucleus of the amygdala and the bed nucleus of the stria terminalis, which are also known to play a role in fluid intake. And so with all of these, I was able to help start to construct a neural circuit diagram underlying this. I'm not going to go in detail today because in the interest of time, but also found some projections to the parabrachial nucleus from the nucleus of the solitary tract, or NTS, and the paraventricular nucleus of the hypothalamus, or the PVH. And more recently, I've been trying to expand that out to look at some of the second and third order projections, some of which extend to the cortex, which leads on to the collaboration I was doing with Dick. So with Dick had been involved in a collaboration with several different people looking at the brain responses in humans, underlying particularly thirst and fluid satiation, amongst other things. And in this particular study, we were quite interested in, rather than just looking at the brain responses to actual thirst and actual rehydration, to look at what would happen to people if they imagined thirst or imagined satiation. So it was one of Dick's great interests, I suppose, the conscious perception underlying these. And so participants were trained under quite a strict protocol in terms of when they needed to imagine being thirsty, and then they needed to imagine drinking. And once they'd been trained up, they were put in an fMRI or an MRI scanner, and their brain was scanned for their particular brain responses. And I've just showed a figure from this article, but a lot of the brain regions that responded were very similar to the brain regions that were shown when people were actually thirsty or actually drinking. So regions such as the anterior mid-cingulate cortex, the anterior mid-insula, the inferior and middle frontal gyruses, for example. And so this suggests that imagining thirst actually activates similar brain regions to thirst itself, and that the experience of thirst can be dissociated from the physiological stimulus, and that the network of brain regions involved in subjective thirst incorporate regions involved in drinking behaviour. So I think it sort of did carry on from a lot of Dick's interests in primordial emotions. And just lastly, and I'm not going to go into detail about any of the experiments for this, but I just wanted to show that I'm also interested in studying the neural circuitry of salt appetite. And with Michael McKinley, I've been looking at stress-induced salt appetite. So several, actually I'll just go to the next slide, several papers from about 40 years ago and going on that Dick was involved in showed that if you either stress an animal or inject a stress-related hormone, ACTH, it'll increase sodium chloride. And you can see in the bottom panel there, there's increased sodium chloride and some water. So I'm interested in using a lot of the techniques which I showed you today to actually tease apart the neural circuitry, particularly underlying stress-induced salt appetite. And so with that, I'd just really like to acknowledge Dick. He's been a great mentor, and I've really enjoyed collaborating with him over the past 5 to 6 years. And it's been wonderful, I suppose, seeing how he interacts with people and forms collaborations. I'd like to also acknowledge the other people on the team listed there, Richard Parmenter and others at the University of Washington, Andy Lawrence here at the Florey, and others, and some of my students, and my funding. And thanks very much.
Thanks very much, Phil, for sharing that excellent work with us. I think it really shows us the evolution of neuroscience over the last decade or two. It also tells us something about Dick, that is that he understood that evolution, and he was prepared to speculate, often speculate over that glass of wine. But in addition, he was drawing people into the fold, the new generation, basically, the people who had the vision of basically establishing new techniques and applying those techniques to actually get the evidence. speculation to evidence, as you've seen from Phil's talk, definitively understanding how neural networks work, and from there can be both innovation and application for benefit for people for the future. I think that Dick understood that. So now let's move on to our next set of speakers, and they are Professor Clive May and also Dr. Lindsea Booth. Now, basically, they're going to talk to us about another aspect of Derek's scientific legacy pertaining to the experimentation on sheep. So I understand that work commenced with Dick's team establishing a parotid fistula to further studies on electrolyte balance, and also adrenal gland autotransplantation to study the hormonal control of salt retention, in particular studies focused on aldosterone. Now, Clive May established the preclinical critical care unit at the Florey and is a renowned expert in integrative studies of the pathophysiology of critical illness using the sheep as an animal model. And Dr. Lindsea Booth works alongside Clive and is one of our rising stars, having recently been awarded an Alan and Maria Myers Research Fellowship. So welcome, Clive and Lindsea. Thank you.
Thanks. Well, thank you, Trevor. It's a great honour and pleasure to be invited to speak in memory of Dick. And I and Lynn both work in the preclinical critical care unit. And as Trevor said, this is a sheep lab where we do translational research into experiments on sheep in collaboration with many clinical colleagues. And I particularly like this portrait of Dick, which Professor McCluskey has described so well. And I think it's also worth pointing out in the top right-hand corner, there's a ballet dancer there, which is reflects his interest in ballet and his obviously his wife's great skill in that area. So The Sheep Hilton is this wonderful book that really describes Dick's evolution from his time at the Royal Melbourne, the studies he did there, and the development of the Florey as a research institute. And as you can see, it's got an introduction by Robin Williams. So this was a description of a patient Dick was dealing with when he was in his early 20s, and this was in 1947. I think this patient has been mentioned a number of times before, but this patient, Reynolds, had had 3 surgeries, had a fistula, and had enormous electrolyte imbalance. And at that time, there was remarkably little understanding about how the electrolytes in the body were controlled. And Dick really began to try to understand this in these patients. And he did that by taking this question into the sheep as an experimental animal, where he used the sheep with a parotid fistula. He then took the discoveries that he made in these animals back into the clinic, where they dramatically improved the treatment of these patients. And I think it's also worth remembering at this time, they really didn't have any facilities to measure these electrolytes quickly. You took a sample, you sent it off to a lab, and 2 days later the results came back. And by that time, the patient would be considerably worse, whereas you needed an instantaneous measure. And they, in fact, ended up ordering a flame photometer that was designed for measuring minerals in soil, and they redesigned the protocol, and they used that and were able to make these amazing advances. So our philosophy in the lab has really been somewhat similar. We take clinical problems that our clinical colleagues have, and we interact with clinicians in a number of hospitals in Melbourne, and we then develop animal models, and we then take our findings back to the clinicians. And this gives you a list of a number of the projects that we've got going in the lab at the moment. Multi-organ failure and septic shock that I'm going to talk about. Then Lynn will talk about autonomic dysregulation in heart failure. We also have projects on cardiopulmonary bypass, epilepsy, stroke, and Parkinson's disease. And in all cases, our aim is to understand the pathophysiology, to develop new treatments and devices and biomarkers, and then with our clinical colleagues, translate these back into the clinic. And you can see the team, recent photo of the team with some of our clinical colleagues and Florey collaborators there. So, as I said, I'm going to talk about sepsis. So this is a life-threatening response to an infection. It can lead to organ failure and death, and it still remains the major cause of death in intensive care units. And when you think back at the work Dick was doing, he was really doing intensive care medicine. He was doing it at a time before it was a speciality. The first intensive care unit in Australia was at St Vincent's in 1961, and he was working in the late 40s. The statistics about sepsis are incredibly concerning. When you look at a worldwide level, in 2017 there were nearly 50 million cases, 11 million deaths. Half these cases were children. 3 million children under 5 die of sepsis. And overall sepsis accounts for 20% of world deaths. And as you can see, in Australia, again, a large number of cases, and even if you're in the best ICUs in Australia, you have a 30% chance of death if you have sepsis. So at the moment, if you go into an ICU with sepsis, they'll give you antibiotics. They then give you fluid resuscitation and vasopressors to support the circulation. These patients have got acute lung injury, so they're put on ventilators, so they have to be sedated. They pretty much all develop acute kidney injury, and in severe cases, they then have to have renal replacement therapy. So all these treatments do is support you, and they hope that if they support you for long enough, you eventually get better and you survive. But as I said, 30% of patients don't. So there are no treatments that reverse the deterioration in clinical state and organ dysfunction in sepsis. So this is a massive unmet clinical need. So like Dick, we took this problem back to the sheep, and we've, over the last 30 years, developed a whole series of complex techniques using modern technology and complex surgical techniques to instrument these animals. So we can put catheters in to measure arterial pressure and heart rate and take blood samples and infuse drugs. We put sensors into the brain to measure brain tissue perfusion, the levels of oxygen and temperature. We put probes on the heart and on the kidney so we can measure blood flows. More probes into the kidney so we can measure, again, oxygen and tissue perfusion. And in some animals, we put in microelectrodes so we can also record the sympathetic nerve activity to the kidney and to the heart. So we're really interested in a sort of integrated approach to understand how the neural and humoral mechanisms are controlling all these different organs, and we can monitor organ function and cardiovascular function on a minute-to-minute basis, 24 hours a day, 7 days a week, in a conscious, alert, awake animal. And what we then do is we induce sepsis in these animals by infusing live E. coli. And what's really important about this model is it incredibly closely replicates the human phenotype. So we get a decrease in blood pressure, increase in heart rate and cardiac output, acute kidney injury, acute respiratory distress syndrome, they get hyperlactatemia, they get febrile, their body temperature goes up, and also they show sickness behavior. So what we have done is this is a sort of culmination of 20 years research in sepsis. We have examined the effect of treatment with what we call a megadose of intravenous sodium ascorbate. And just to remind you, sodium ascorbate is the sodium salt of ascorbic acid, which is vitamin C. So we're giving 150 grams of vitamin C, sodium ascorbate, intravenously into a 40 kilogram sheep. So this is a massive dose that has not been used previously. So this shows you the clinical state during sepsis. These animals look much like you would do if you've got a bad dose of flu. They lie down, they're not alert, they don't want to eat and drink. They've got a high temperature, and you can see in the graph here, this is the basal temperature, and this is during sepsis. You can see temperature goes right up, and it stays at this very high level during the experimental period. At the end of the period, we give them antibiotics, and we stop the infusion of E. coli, and over a 48-hour period, the animals then recover. What happened when we infused sodium ascorbate is the most remarkable thing I've ever seen in 50 years of research. These animals, within 3 hours, were standing up as if they were totally normal, healthy animal. So they were looking well and alert, they were eating and drinking, and remarkably, over that 6-hour period, their temperatures, body temperatures, returned back to normal. And I would like to acknowledge Yogesh Lankadeva, who has played a major role in this work, and also congratulate him. He's recently been the lead CI on a 4.9 million MRFF grant that's going to enable us to continue this work and continue into clinical trials. So if we look at the effects on blood pressure in this left-hand graph, as I said, blood pressure drops dramatically in sepsis. We then give fluid resuscitation. As is given clinically, we get a slight increase in blood pressure, but it's not maintained. So we then have to give this drug noradrenaline to maintain blood pressure back to a normal level, as is done clinically. And what you can see, in order to do that, we have to ramp up the dose of noradrenaline that we give, because as the animals get sicker, they become less and less sensitive to noradrenaline. When we treat, in the group that we treated with sodium ascorbate, you can see that blood pressure was maintained extremely well during the experimental period. And this was done despite the fact that we reduced noradrenaline levels to 0 in most of the animals. So this indicates that the cardiovascular system and the responsiveness to the endogenous vasopressors is returning back to normal levels. As I said, a classic hallmark of sepsis is acute kidney injury, and this is indicated by a decrease in urine flow and an increase in plasma creatinine, which is the clinically used marker showing that the kidneys are failing. And you can see that there is a large increase in plasma creatinine, and that's maintained throughout the experimental period, and that only recovers when we stop giving the E. coli and give antibiotics, but even then it takes 48 hours to recover back to normal. When we infused sodium ascorbate, you can see there is an immediate massive increase in urine output, and plasma creatinine drops and returns back to normal, indicating that renal function has completely recovered. As I said, we also measure the oxygen levels in both the brain and in the kidney, and you can see there are these dramatic decreases in oxygen in the brain tissue and also in the renal medulla, which is the inner part of the kidney. And again, these low levels are maintained throughout the experimental period. As soon as we started the infusion of the sodium ascorbate, you can see there's a big increase in the oxygen levels in the brain back to normal. And as the oxygen in the brain returned to normal, so did the animals wake up, become alert, standing up and starting moving around. And in the kidney, again, as the oxygen level returned back to normal, so did renal function restore itself. So our clinical colleague, Ronaldo Bellomo at the Austin Hospital, has just finished a phase 1A clinical trial. in which he gave 60 grams of intravenous sodium ascorbate to septic patients. He had to give a very, very much lower dose, about a quarter of the dose that we give in our sheep, because for safety reasons he wasn't enabled to give the very high dose that we give. But this trial has reached its primary outcome of an increase in urine output. They also, as you can see in this graph here, the red is the treated group. They also showed a decrease in noradrenaline requirement to maintain blood pressure, as we saw in the sheep. And these patients also had an improved sequential organ failure assessment score. So this is a score of gives an overall integrated view of their organ failure, and there was an increase in improvement in their organ levels of organ failure. So in septic patients, intravenous sodium ascorbate at a much lower dose had qualitatively similar, although reduced, effects to the very much higher doses that we're giving in sheep. And we're now going to move on to some clinical trials with high doses. So what I've shown you is that in established ovine sepsis, treatment with intravenous sodium ascorbate was safe. It restored the clinical state and improved cardiovascular state and reversed the organ failure. In patients with septic shock, a lower dose had qualitatively similar beneficial effects. And now we're planning to move on to a phase 1b trial with a much higher dose that's going to be given over a 24-hour period in 30 patients. And if that's successful, we're going to move on to a multi-phase phase 2 trial with 300 patients. So we hope that megadose sodium ascorbate will transform the treatment of septic patients worldwide. It'll reduce the degree of organ dysfunction in recovered patients, because this is a major problem with patients who've had sepsis. Many of them have long-lasting brain and kidney injury. It'll reduce the costs in ICU, which are currently extremely expensive, and we hope that it will save many lives. So I'd just like to finish with what I think is a very apt quote from Isaac Newton in a letter he wrote to a scientific colleague. And clearly the work that we have done and we have developed in the sheep lab has really depended on Dick's vision in using sheep as an experimental model. And we have a group of incredibly talented younger people who will now, I'm sure, continue Dick's legacy and take this on to bigger and greater things. So thank you very much. Now, Lynn.
Thank you, and I'll just finish the last half of this presentation, and I'll try and go quickly so we can get to morning tea. So I'm going to be talking to you about the sympathetic nerve activity in our large animal model of heart failure, specifically looking at renal denervation as a treatment, a potential treatment. So why do we study heart failure? Well, an estimated 480,000 Australians are living with heart failure, with an additional 60,000 diagnosed every year. This comes at a cost of an estimated $2.7 billion per year in healthcare costs. So despite current treatment options, heart failure has a high mortality rate, with about 1 to 2 or 1 to 3 people, 1 in 3 or 1 in 2 people dying within 5 years of diagnosis. So it is well established in heart failure that there's an increase in sympathetic drive to the heart and a decrease in parasympathetic drive to the heart. Now, this autonomic dysfunction is associated with increased mortality and morbidity. What's important is what drives these changes is not completely understood, and this is where we're really interested. This is where our research is focused. So Clive, with others, developed the heart failure model in sheep in our lab. So it's an pacing-induced heart failure model, and we say that sheep are in heart failure when their ejection fraction, so how much blood the heart is pumping out, falls below 40%. And this is the equivalent of mild heart failure in humans. So I'm just going to show you some data here. I can see the marker, cool. So we've got normal sheep in this column here, heart failure sheep on this side. I'm showing arterial pressure at the top and central venous pressure below. As you can see with heart failure, there's a slight drop in blood pressure and an increase in central venous pressure, which is seen in human heart failure patients. We also directly record sympathetic nerve activity in these animals, and this is looking at the renal sympathetic nerve activity, so the nerve activity going to the kidney. We've got raw activity here and integrated activity underneath. I'll draw your attention to the raw activity. You can see that nerve activity occurs in bursts, and these bursts occur at the same time within the cardiac cycle. So this is very typical renal sympathetic burst. In heart failure, there's a slight increase in renal sympathetic activity, but it's pretty high in normal animals, so there's not a lot of capacity to increase. Underneath here, we see cardiac sympathetic nerve activity, and this is in a normal animal. You can see the burst rate to the heart is much lower in normal animals, and this is significantly increased in heart failure. Prof Denton was very interested in this work, and indeed he was part of the team that was successfully applied for an NIH grant back in 2004 to establish this model further and to study its potential. And this is just one of the papers from the work that was done and funded by that grant. So now I'm just going to take you through just a little bit of data that I've generated, looking at a potential treatment in heart failure using this heart failure model. So catheter-based renal denervation appeared about 15 years ago as a treatment option as a trial treatment in patients with resistant hypertension. So this is a relatively non-invasive method. This is the catheter here. It's guided into the renal artery, and then the nerves are ablated from inside the artery. So the proposed mechanisms of action include removal of the efferent sympathetic nerves and the afferent sensory nerves. So by removing the efferent sympathetic nerves, there's a decrease in renin release, renal vasodilatation, and an increase in sodium excretion. And by removing the sensory afferent nerves via central spinal pathways, there's a potential to decrease global sympathetic activity. So both of these things could be beneficial in heart failure. And indeed, catheter-based renal denervation has been trialled as a potential treatment. Back in 2013, they did a safety trial in heart failure patients, and they showed no adverse effects and actually an increase in the 6-minute walk distance in these patients. There's also evidence that there's a reduced potential for arrhythmias after renal denervation in heart failure. So the aim of my study was to determine the effects of catheter-based renal denervation in sheep with heart failure. So this is just a couple of fluoroscopic images from the experiments. We've got the kidney up here, and this is the denervation catheter shown here. So we use the Simplicity catheter, which is the same catheter used in patients. And we do 6 ablations per kidney. The ablations start as close as possible to the kidney, because this is where we know that the nerves are close to the renal artery. So the studies were done in sheep with pacing-induced heart failure. We had a group with renal denervation and a sham group, so where the catheter was introduced into the renal artery, but no ablations were performed. We measured heart rate, blood pressure, cardiac sympathetic nerve activity, and an indirect measure of parasympathetic activity. So I'm showing you here some data. This is the mean arterial pressure and heart rate at a time point before any intervention, 1 week and 6 weeks post intervention. Renal denervation is shown in blue, and the sham is shown in the open circles. So you can see in mean arterial pressure, 1 week after denervation, there was a slight drop in blood pressure in the renal denervated group, but this had come back to baseline by 6 weeks. If you look at heart rate here, we saw no change in heart rate 1 week after denervation, but interestingly, 6 weeks after denervation, there was a drop in heart rate in the renal denervated group, suggesting that we're seeing maybe a change in the autonomic balance going to the heart. So then we wanted to look at indicators of parasympathetic drive. So what I've shown here is a baroreflex curve, a heart rate baroreflex curve, and I'll just talk you through it. Heart rate is on this axis here, and systolic blood pressure is along here. So we use vasoactive drugs to increase and decrease blood pressure and look at the reflex changes in heart rate. As you drop blood pressure, there's a reflex increase in heart rate, and this was not affected by renal denervation, which are shown in the blue again. However, when we increased blood pressure, we saw this decrease in heart rate that was greater after renal denervation. And what I need you to know here is that this reflex is actually blunted in heart failure. So this is not a normal decrease in heart rate in response to an increase in blood pressure. So this is an improvement in the parasympathetic drive. This indicates an improvement in parasympathetic drive after renal denervation. The other thing we can look at to get an indication of parasympathetic drive is heart rate variability. The higher the heart rate variability, the higher the better it is for the heart. So here we're looking at variability before and after either renal denervation or sham. You can see here we get a slight increase in heart rate variability after denervation, as opposed to a slight decrease after sham, again suggesting an increase in parasympathetic drive. So as I showed you before, we can directly record cardiac sympathetic nerve activity in these animals, and this is the renal denervation group here as opposed to the sham. And the sham has that very high increase in cardiac sympathetic nerve activity that I showed previously, and after renal denervation, we saw a significant drop in this cardiac sympathetic nerve activity. So we quantify this by looking at the bursts per minute, and after renal denervation, there was a significantly lower level of cardiac sympathetic drive. Just to convince you that we actually denervated the kidneys, we looked at noradrenaline content of the kidneys, renal denervation, and sham, and in the denervated group, there was a significantly lower level of noradrenaline. If it was surgical denervation of the kidneys, we'd expect this much closer to zero. So catheter-based denervation is partial denervation of the kidneys. There's still some nerves going to the kidney. So in summary, a drop in heart rate, increased parasympathetic drive, and decreased cardiac sympathetic nerve activity suggest that renal denervation may positively alter the long-term autonomic control of the heart in heart failure. And just a few final words. As Clive pointed out, we're part of the preclinical critical care unit. This is us at our Christmas party. As a group, we are focused on working with clinicians, engineers, and other scientists to keep our research goals clinically orientated and cutting edge. And we are honoured as a group to carry on Prof Denton's legacy of the sheep Hilton, and perhaps one day grow it to the Ritz-Carlton. Thank you.
Thanks very much, Lindsay and Clive. And I think you can see how integrally important those results are. It's really exciting research. And I think if we reflect on sheep studies at the Florey, the chronology is triphasic. Initially it was seen as absolutely integral, then we went through a phase when it wasn't necessarily seen as avant-garde. But I think the Florey and the University of Melbourne have now caught up with the vision of Dick Denton and also Clive May and others, and recognise that a cutting-edge campus such as at Melbourne needs to engage in large animal research, because it's important in terms of translation. There's a big gap between rodent or mouse studies and studies in man. It's horses for courses, but we recognise it has a role, and that's been recognised by our partners as well, such as the Bionics Institute near Barnica. And in addition to the critical care studies, there's a lot of medtech work which is being undertaken in the sheep facility now, related to vagal nerve stimulation, multi-electrode arrays for epilepsy monitoring, the stentrode research, which probably many of you have heard in relation to Tom Oxley's work, emanated from the sheep studies as well. So we see it as absolutely integrally important to the Florey's future, once again consistent with Derek Denton's vision. Now, we're going to put the presentations on pause. We're going to enjoy a short morning tea in the foyer area. We had planned for 20 minutes, but we are behind schedule, so I just encourage people to come back at 25 to 12. Thanks very much, everyone. Welcome back. If you could take your seats, please. So it was good to see everyone taking Derek's lead and having long, productive conversations over, in this case, non-alcoholic beverages. We couldn't, unfortunately, give you that red wine, but we do have to get the show back on the road. And I now would like to welcome Professor Brian Oldfield to speak. Brian trained at Monash and McGill universities before returning to Australia to spend the formative years of his scientific career here at the Florey, during which time he shared multiple important publications with Mike McGinley and with Dick himself. Mike was actually one of Dick's closest colleagues. Mike won't be talking today, but is here very much to be part of an integral part of the day. Brian subsequently returned to Monash in 2005 after really cementing his focus on the interrogation of the neural pathways controlling thirst, hunger, and ingestive behaviour. And he's had a continuing focus on understanding appetite, energy expenditure, and body weight. And over the last decade, in addition to his research interests, Brian has broadened his focus to take leadership roles in agencies and societies to improve the care and management of those living with obesity, most obviously recently in Southeast Asia. Brian has elected to entitle his talk "From Cellular Sensing of Thirst to the Nature of Consciousness." Please welcome Brian.
Well, thanks, Trevor. Look, for a start, I'd like to say that I see this opportunity to speak at Dick's Festschrift as a great honour, and I would like to thank those that suggested that I take part. I was employed by Dick in 1985 on the back of following a recently conducted quinquennial review, remembering at that stage the Florey was one of 5 block-funded institutes in the country. And the suggestion from that review was that the Institute needed more strengths in neuroscience, and particularly in neuroendocrinology. And I guess that wasn't any surprise, given that the head of that examining committee was Professor Roger Guillemin, renowned neuroendocrinologist and recent winner of the Nobel Prize for his work on hypothalamic peptides. So I guess for me, it was right place, right time, and certainly one of the most pivotal times in my career. And no small part of that was the example and influence of Dick Denton during my time at the Florey. Now, the title of this talk that Trevor has given you may appear a little presumptuous, and I think it is, but hopefully you'll see where I'm going with this. I'm going to try and bookend this presentation with 2 separate and disparate interests, and they're shared interests with Dick in some ways. On the left-hand side, I'm going to start by talking about the cellular sensing of thirst, work that I did very closely with Michael McKinley and others at the Florey in our early days. And then, for the most part, I wanted to talk about the nature of consciousness, or at least as it applies to the effects of psychedelics in anorexia nervosa. And this is some of the work that my lab at Monash has focused on more recently and has been taken on very much, as I'll point out during the presentation, by Claire Foldy, who is pivotal in this work. I won't be talking about the neurocircuitry of energy balance, the physiology or the physiological regulation of brown adipose tissue, or the mechanistic underpinnings of bariatric surgery, which is the thing that I would normally talk about in these presentations. So with that left-hand bookend, a few years before I arrived at the Florey Institute, Michael, Dick, and Richard Weisinger, on the basis of some very good physiological experiments, suggested or proposed that the osmoreceptor would exist, or that should detect changes in osmolality in the circulation, coincident with changes in dehydration. But this would be detected, this change in osmolality would be detected by a cerebral osmoreceptor, and Michael suggested that that cerebral osmoreceptor was likely to reside behind the blood-brain barrier, or in an area without a blood-brain barrier, sorry. So What happened then was that with the freedom that we had at the Florey, which was, I think, fair to say, established and encouraged by Dick, we pushed the idea that was introduced by Phil, and thank you for that the elevation, this is a very new concept at that point, that the elevation of the protein product of the CFOS proto-oncogene could be used as a marker of neural activation. And we used that to visualize for the first time the cerebral osmoreceptor. And this is shown here in this publication with some of the authors, Michael, pivotal in this work, and even though Dick's name is not on it, he was very much involved in the process. You can tell it's an important experiment because in the photograph you can see 3 of the authors on the paper supervising the mounting of brain sections that and Michael actually orchestrating this with a pencil, which is... But the end result was this sort of, in the histological picture, you can see that the FOS response, shown here as a brown reaction product, neatly outlining this group of osmoreceptive neurons for the first time in the dorsal aspect of this area called the OVLT, organum vasculosum of the lamina terminalis. Now, I would also say that I remember that this initiative came out of the very lively and supportive and collegiate lab meetings that we had amongst this group that included all of the people on this paper and Dick. And I think this was I think in the heyday for me of that sort of collaborative arrangement, and it was a very productive time. So the right-hand bookend. This is where I'm going to settle for the rest of the talk. But it's in deference to Dick's interest that's already been mentioned a couple of times in the nature of consciousness and self-awareness. Now, and this is highlighted in his book, The Pinnacle of Life, which I think was the last of his published books. I think if you take nothing else out of this slide, you can see that it highlights the fact that many of us recognize and we're in awe of, is that Dick had almost unparalleled ability to establish and then draw on his networks. And here, if you can see in the fine print, his involvement in this book with discussions with Miriam Rothschild, a preeminent studier of fleas in Britain, who had some views on consciousness and member of the iconic Rothschild family, and also the Australian Nobel laureate John Eccles. And I think that was very typical of the way that's already been pointed out by Professor McCloskey and others of the way that Dick operated. In a review of that book, it was stated that Professor Denton is a materialist, believing that consciousness, or the primary sensual consciousness, is a function of the brain and its circuitry. So Dick's view of consciousness was quite pragmatic, and perhaps a little removed from some of the more philosophical approaches to consciousness, which may be an awareness of being. But Dick, I guess, was more focused on those parts of consciousness that could be tested empirically. And this opens into considerations of the so-called easy problems by those studying consciousness related to attention, perception, and interoception. And I think that fits very well with the way we have utilized psychedelics to understand the mechanisms underpinning anorexia nervosa. Sorry. I'm struggling with, I have medications for a cold and it's drying me out. So just in terms of anorexia nervosa, most of you will be familiar with the nature of anorexia nervosa. It clearly involves food restriction, hyperactivity, both of which are voluntary, and an extremely low body weight. I've referred to Kate Moss here, which is an unusual reference in this setting, I guess, but this was her at the height of her supermodel career, where she famously said, "Nothing tastes as good as skinny feels." And when she said that, I thought that was quite clever, because it actually encapsulated and showed to the world what it was like to feel anorexic. I also, I was a little enamored of that and used it as the title of an NHMRC application that I'd like to say was successful, but they didn't share my enthusiasm for that. But you should also remember, and I don't want to make light of this, because anorexia nervosa, we have to remember, is associated with the greatest mortality of any psychological illness. If you confound that by just focusing on females between 15 and 24, then there is 12 times greater mortality than death in that age group from any other cause. So this is a very significant problem that has no clear medical treatment. We now know that its causes are genetic, physiological, neurobiological, psychological, and environmental. We know quite a lot about that. We realize that those causes result in disturbances in motivation, cognition, interoception, and I'll be talking a lot about reward processing, or some about that. And these things are manifest in aberrant eating, physical activity, low body weight, and a generalized anhedonia. This is an anhedonia, or an unease with pleasure that is not only associated with food, but in all aspects of life. So if we consider the neurocircuitry that underlies this problem of anorexia nervosa, and I should point out that this is true in acutely ill or weight-recovered patients, which indicates a biological basis for this. First of all, the first element is there is increased activation of prefrontal cognitive circuitry. So there's an overactive part of the brain that is involved with excessive cognitive control. And this makes sense if you think about the amount of rigidity that's required for somebody living with anorexia on a starvation diet for weeks, months, and possibly years until they possibly succumb. So that involves an extraordinary amount of rigidity. That's the first hallmark. There is also, working against that, a decreased activation of not only the primary taste cortex, but these mesolimbic reward pathways. So there was a reduction in the reward. And this results in an altered reward valence. And by valence, I mean the motivational value that is ascribed to certain food-related cues. We should also remember that there's a disruption in the way that anorexic patients deal with this interoceptive information in these food-related cues through reward and cognitive pathways. So they are, unlike healthy controls, not influenced by their fed or fasted state. That's irrelevant to people living with anorexia. So what we need to do is to re-establish this balance of reduced reward and elevated cortical control. To do that, we've focused in my lab over the last at least 20 years on an activity-based anorexia model. This is probably the most important or the well-studied of all of the models of anorexia. And it's very difficult to have a true model of anorexia nervosa in an animal. But it involves free access to running wheel on one hand, combined with 90 minutes or a restricted time availability to food. So they can eat as much as they like, but only for 90 minutes. And this results in a recapitulation of the core features of human anorexia. It has rapid body weight loss, hyperactivity, and hyperphagia. It also causes changes in metabolism and hormones that are very much akin to the anorexic situation. And this is the sort of result that you can see after the combined effect of these two parameters. And shown here in black, you can see that in the first of these 10 days of the experiment, that the animals they dramatically lose a large amount of weight. And in fact, in the first 6 or 7 days, they are likely to get to a point where they lose an amount of weight that puts them in danger. And so we have to ethically take them out of the experiment, and this happens at about 20% weight loss. It's also important to remember that in terms of this paradigm, you have to have both of these things together, because if you lock the wheels and just use the restricted availability of food for that time period, they lose weight for the first couple of days because they don't realize you're going to take their food away. And then once they do, they maintain their body weight pretty well at a standard level. So So I'm thankful to Phil for outlining the basis of chemogenetics, sorry for the spelling error in the first, manipulation of these circuits that change the presentation of anorexia, because subsequently, and very much in conjunction with Claire Foldy, we've been doing these studies where we've manipulated, firstly, these reward pathways by enhancing the activation of reward pathways, specifically those, using DREAD technologies and chemogenetics, and we've been able to rescue animals and prevent anorexia nervosa. And in a contrary fashion, in a subsequent publication, we've been able to inhibit the overactive cognitive centers that create this inflexibility or cognitive rigidity. So this leads me to, I guess, a sliding doors moment or a light bulb moment for me, because about 3 years ago, I was given this book by my children, and I read it over the Christmas break. It is How to Change Your Mind, the New Science of Psychedelics, by Michael Pollan, best-selling writer, teacher of writing at Stanford and so on, with the subtitle, What the New Science of Psychedelics Teaches Us About Consciousness. Now, I was absolutely intrigued by this book. It's a great read if you haven't read it. And Michael Pollan came out to Australia and brought out by the Weller Foundation, and I was lucky enough to spend time with him. We have a little bit in common, but at some point in that book, it pointed out that psilocybin, the active ingredient in magic mushrooms, at high doses leads to a reduction in cognitive rigidity, or in other words, an increase in cognitive flexibility. And so this resonated with me straight away. And so what happened on the basis of that, within a week, Claire Foldy and I had initiated a process of bringing psilocybin for the first time into Australia for experimental studies. And we were on the path to doing this work. And I have to say, in case I don't say it enough, that Claire has taken this ball and run with it. This is her now in her newly established group of her own with independent funding and so on. I also want to just reflect for a moment on the fact that a lot of the fanfare about psychedelics and their potential to treat a whole range of psychological illnesses seems like it's for the first time, but it's actually a second coming. Many of you will appreciate that in the 1960s, psilocybin was fairly widely used in clinics across the United States for the treatment of alcohol and nicotine addiction, and treated very successfully. And Sandoz, in fact, started selling psilocybin. The process, which is interesting, but for another time, was in some way derailed by Timothy Leary and his colleagues as part of the Harvard psilocybin project, where Leary felt fairly destructively that this should be in the domain of the general public, and that it would enhance our social interactions to that point. And so he started a movement that was essentially blocked by conservative governments at the time. So what it did was it led to the Controlled Substances Act, which outlawed all research onto psychedelics for 30 years, as well as outlawing the substance itself. So it wasn't until nearly the turn of the century that we started to look again at the pharmacology of psychedelics. I'm just going to move through this fairly quickly, but thereafter there were started to appear a range of different studies around the world using psychedelics for clinical conditions, and culminating in 2018 in the US FDA designating psilocybin as a breakthrough therapy. This was a big step for the FDA for depression. And the use of psilocybins and the claimed potential of that has escalated ever since. And I would just put a cautionary note on that, and we have to be careful of the hype. This is not the first time we've seen it, because in fact a very similar Newsweek article heralded the effect of Prozac as the greatest answer to depression. And so I think we have to be a little bit careful, and the message that Claire and I would have is that we need to focus our attention on on understanding the neurobiology, mode of action. And part of that is developing and testing psychedelic analogues that still have therapeutic value, but don't have the hallucinogenic properties. And we have some connection with a Danish group that is investigating that. And that leads to what we see as a virtuous cycle where we understand fundamental neurobiology, we can investigate cognitive flexibility through use of psilocybin, and via rational pharmacology, hopefully come up with some ideas on therapies. I'm just going to finish with a couple of slides on the psilocybin administration that Claire and her group have done. And this is in conjunction with the activity-based anorexia model, where you precede the paradigm, the ABA paradigm, with a single dose of psilocybin. Or a controlled situation. And the first thing you notice is the susceptibility to the ABA paradigm, where all of the animals would potentially die, is that 50% of them are resistant. And this is shown in two different ways here, with the animals shown in the dashed lines not reaching this cutoff point ever. So they're actually resistant to the anorexia-inducing ABA. The final data slide also takes that information and starts to investigate it a little more. And this is Claire's forte, I think, in advanced behaviour. And what she's done and her group have done is that they've trained rats in this operant training program to press or to poke for a reward, a food reward, on one side of this apparatus. And they do that until they're very good at it, and then they switch it to the other side. And so there's a change in rules. And these plots on the side just illustrate that those animals that have received the psilocybin in blue are much more flexible in understanding the new rule. They perform less perseverative errors, those continuing errors where we go back to the old rules. So they've actually they've had cognitive flexibility induced. So I would then finish this by saying that I think, or the scientific part, by saying watch this space. I think we need to focus on the neurobiological impact and the structural underpinnings of the way that psilocybins work in the brain. And we also have to think about this idea of developing non-hallucinogenic analogues, because the tripping part of psychedelics, which may be 6 to 8 hours, I think is contrary to ease of use in the clinic. I would like to, and I would also ask you to follow Claire Foley. I wanted to finish up, and I was surprised in a way that this came up with Professor McCluskey's opening, by asking this question of what is the common language of scientific research, and how does this relate to Dick? And this came out of, and this was a reference from Professor McCluskey, that I've been intrigued by this since I heard John Funder talk about this 35 years ago. And he gave an hour-long lecture of what is the lingua franca of scientific research. What's the common language? And he came up over that hour, and I will take 30 seconds, that it may be fun. I mean, Professor Mendelsohn said to me once that if you are not having fun, you shouldn't be doing research. Competition ego, I'm so sure, challenge, obsession, I think that's true. Mystery and intrigue, yes. Inquiring, curiosity, I doubt status and money, but if that works. But John Funder and I agree that the lingua franca of scientific research is a feeling of amazement and admiration caused by something beautiful, remarkable, or unfamiliar. And I can honestly say that I have never seen a scientist driven more completely over as long a period of time by wonder, and is fueled by a lack of constraint, whether that be intellectual or financial, than Dick. And it was an honour to be close witness to that process, so thank you.
Thanks very much for those insightful comments, Brian. And I'd like to add to it as well, and I think that you demonstrated that in terms of relevance and connectivity, nimbleness, to be able to at one glance look at a particular nucleus within the hypothalamus but then to see the relevance of that to global issues in terms of eating disorders and one's activities being relevant to help solve other people's problems. So I think that's another aspect of being a researcher. I think you also drew into the discussion at the start this perspective of Dick's understanding of the scientific diaspora. Wanting to make connections, wanting to draw people into the environment, basically, who had something to give, and he saw that obviously in you. And I think that we at the Florey need to see that as well in terms of the scientific diaspora and its connectivities to other institutions in Melbourne, basically. And it's great to see friends and colleagues from Monash and other medical research institutes here today as well. And I think hopefully today it just helps us to enhance those connectivities, because at the end of the day, although the Florey was Dick's greatest love, he absolutely appreciated that at the end of the day science went beyond institutional boundaries. And just finally to reflect upon this connectivity between instinctive behaviour and mental health. We're also very appreciative of that here at the Florey now. We have mental health initiatives which are extending to collaborative interaction with both the University of Melbourne and also Monash, particularly in neuromedicines and embracing, as you've said, psychedelic research and its renaissance. So that said, let's move now to our next presentation, which will be from Professor Gary Egan, who's been director of both the Monash Biomedical Imaging Facility and the ARC Centre of Excellence for Integrative Brain Function. Gary is a renowned expert in biomedical imaging and its use in providing insights into structural and functional neuroanatomy, in other words, brain mapping, at the mesoscopic level, and its application to understanding the pathogenesis of neurodegenerative disease. Gary, before taking up his senior appointment at Monash, had a long and deep association with the Florey, including with Dick and also with colleagues at the Research Imaging Centre in San Antonio. Included in the collaborative research was work focusing on the neural networks driving the consciousness of thirst, ultimately seeking perspectives about the evolution of consciousness from instinctive behaviour. With that in mind, Gary's topic for discussion today, or his presentation today, is going to be Imaging Interoception, a 25-year journey with Derek Denton. Thanks very much.
Thanks very much, Trevor. It's a real honour to be able to present here at the Festschrift in memory of Derek Denton. Derek was an amazing person, and it's great that we're having this day to celebrate his contributions. I actually first met Dick just in the car park outside the Institute, in fact the old Institute, and that was at the introduction of Professor Fred Mendelsohn, then the director of the Institute. And it turned out that Dick was doing experiments down in San Antonio in Texas with a group of people who I was also collaborating with, and we'd been there visiting in San Antonio at different times. And Fred knew that, so he introduced me, and from that was born a fantastic 25-year experience with Dick and collaborators. I wanted to just kick things off by introducing a few of the people and some of the situations. We did a lot of research there. We actually did quite a bit here in Melbourne in the latter stages, but initially it arose through Dick's work at the Southwest Biomedical Research Foundation using the baboon model and working with Bob Shade. You see there in that top left photo. Bob was the scientific director of the Institute there, and the idea that Dick had was to actually image a baboon in a PET scanner, and the only PET scanner in town was over at the Research Imaging Centre at the University of Texas Health Sciences Centre, and that was led by Peter Fox, one of my colleagues and the person who I was collaborating with. So it was a fortuitous connection, which then led on to a huge body of work, which I'll share with you in brief. But the experience there in the United States was, was multifactorial, I guess you would say. And it included, of course, a lot of great fun, as we just heard from Brian. That was a big part of the program with Dick. It also involved important meetings, one particular, which I've got a photo here, at the Howard Florey Biomedical Research Foundation, which was meeting in New York at that time, and you can see here Martin Meyer in the photograph below, where we had the dinner and the meeting, the annual general meeting, together with Fred, in the auspicious environment of the New York Yacht Club. And at that stage, we'd actually lost the trophy back, so it was a little bit of a downer. They had great pride in showing us where the trophy was now repositioned in the glass cabinet in their club. And it was also a joy to share some of that with Dame Maggie Scott. She came over on a couple of trips. And how we did this research was quite like you might call suitcase science almost. We would jump on a plane, myself, and then at a later point Michael Farrell. We would jump on a plane, get down to Texas on Sunday evening. We'd go to the Liberty Bar where Dick would start the networking for that particular visit. Then Monday morning we'd arrive at the scanner, which had been reserved for Dick for 2 solid weeks. He was able to command that time. We would run experiments, 2 a day for the fortnight. That would be 20 subjects. In the afternoons and evenings, we'd be looking at results and reviewing data, and then, of course, going out and enjoying the river walk along the San Antonio River there in San Antonio. So it was a terrific experience and actually incredibly productive, as I hope you'll see. The body of work was around looking at interoception and the neural correlates of that, the neural correlates of the consciousness of these interoceptive processes, and in particular their association. It was a program that started with thirst. It continued on looking at hypercapnia and then actually air hunger. We continued work, some in San Antonio, some here in Melbourne, looking at thermoregulation, and Robin McCallan, who'll speak next, was the lead scientist on that work. We did further investigations of simultaneous processes looking at thirst and pain. We looked then in more detail, inspired by some of the work that Phil has pointed out, to look at circuitry associated with some of the subcortical structures, in fact in the OVLT and the anterior wall of the third ventricle. So we tried to look at the ascending circuitry in the brain and how that manifested and was extinguished through satiation of the processes. We had a little side excursion at one point. We went over to Groningen. I won't say any more about that until I get to the slide, but that was really good fun, looking at satiation processes. And then finally we started manipulating the consciousness experience of thirst in a number of ways. As Phil also said, by looking at imagining thirst, as we had an experiment particularly looking, I guess, at how you could try and mimic different components of the thirst experience, that triumvirate of the sensory input from the mouth, the oropharyngeal processes, and then the gut distension. And so we actually did nasogastric experiments in the PET scanner in San Antonio, where we put water in, we took water out, we made people drink, and we played around with these things. So it was really great fun. But the first study, which was published in '99, I believe, was in fact, I think, a tour de force by Dick. He had approached a number of imaging centres worldwide to conduct what you might think of as the first physiological brain imaging. It might not seem very sort of radical today, but at that time, most of the work done with PET activation scanning and functional MRI later, was looking at cognitive processes and not physiologically driven changes in brain activity. So Dick proposed that. I know he went to Sweden, to a leading facility there, to engage with Per Roland. Didn't quite work out. He then visited San Antonio, as I mentioned, with Peter Fox. And Peter was a bit of a left field thinker and sort of took on this challenge. And when these results were first presented, I think everyone in the room jumped and said, my God, it actually works. We really generated brain activity changes that were measurable with PET cerebral blood flow measures, which were correlated with the processes of plasma sodium concentration increases when individuals were lying in PET scanners and given these infusions of high molality plasma. So it was a really exciting finding, and you can see this paper here, which had a whole series of activation areas along the pregenual cingulate, and then the anterior mid and the mid cingulate cortex were to become the focus of our subsequent investigations. And the paper really concluded that there was a role of the anterior cingulate in the experience and the genesis of thirst, which then led on to a whole series of subsequent experiments. As well as the physiological process, looking at the sodium plasma concentration, we also looked at people's subjective experience of thirst, so we asked them to rate their thirst scores during the experiment. And then we had a profile you see here of the experience that people reported to us during these 7 or 8 PET scans. And then we correlated the thirst score against cerebral blood flow changes in this cohort. And you see it's a very different pattern with the posterior cingulate actually, which now we know as a part of what's called the default mode network in the brain, which wasn't even discovered at that time. And so we identified this area as an introspective part of the brain that was processing people's sense of their experience of thirst. But that same experimental data was able to be analysed to look at the responses in the brain. You see in the top right here of maximum thirst. 3 minutes after people drank, you see a lot of the activity disappeared, but still this pre-genual area continued. But then quite much later, that area of activity disappeared. And the conclusion of that experiment in general was that the consciousness of thirst is in the brain, and we can go hunting for this and explore it further. Subsequently, the work looked at the processes of hypercapnia, so asking individuals to inhale an 8% carbon dioxide air with a mouthpiece, and separately with a face mask, as well as control scans looking at oxygen and just room air. And we're able to establish in that experiment the consciousness of air hunger through a very clever comparison of the same physiological situation of 8% carbon dioxide but inhaled through a mouthpiece versus inhaled through a face mask, with a much less, if you like, strong experience of the breathlessness. And so this data, as you see here on the left, showed a complex area of neural activity, cortical correlates of this air hunger, but again we saw these regions in the cingulate cortex and more in the anterior and anterior mid-cingulate, which were associated with this experience. So the point being that there was no physiological difference in these individuals, but their personal experience of air hunger was different in the two states. And in that paper published in the same year, I think, or perhaps the next year or a year or two later, was actually an overview that Dick put together, which was encyclopedic in a way. He drew on data from many other published studies to map these interoceptive experiences, the processes of consciousness of them, across the anterior cingulate cortex. And that's what's shown here in this paper. And the conclusion there was that this theory of phylogenetic origin of consciousness arose from primal emotions engendered by an immediate threat to the existence of the organism, which was a lovely way of summarising the encyclopedic approach and the panorama that Dick was investigating at that time. He also constantly said to us, it's no good working down in the weeds. We've got to hunt the big game when we're doing this sort of research. And so it was always an exciting time with him. This work, plus some other later work, was summarised in a paper in 2008 in Consciousness and Cognition, where he then recapped some of this work and then added to it the next step, which was to really look at the satiation process. Having established these brain areas that were involved in the interoceptive experience, the consciousness of them, once you satiate either your thirst or air hunger, what happens to those areas and how quickly can we identify and make their responses and diminution specific to that process. And so we did an experiment out at the Brain Research Institute at the time, led by Graham Jackson, to again infuse in a number of individuals hypertonic saline, and with the greater temporal resolution of functional MRI, as shown here in this time course, we're able to look at signal increase, again in this pre-genual cingulate area, increasing in time with a hypertonic infusion, and then at the point of drinking, a diminution of that signal, and at the point of a subsequent drinking, a diminution of that signal in further. So we were looking at this point now to capture that moment of satiation and map those brain areas that very, very specifically associated with the loss of the consciousness of that, that physiological process. So that was very exciting. We then, together with Robin McCallum and colleagues, did a first experiment over in San Antonio. Was led by John Johnson there, who worked in the University of Texas. He fortunately had access to one of the original Apollo space suits, which was plumbed and could be then set up to be able to cool or heat a person as they were lying prone in a scanner. And so this is what we did. That photo is actually from the subsequent experiment done here in Melbourne, but we did a PET experiment in Texas where we looked at essentially the skin and then the body core temperature of the individuals who were lying in a scanner being initially cooled down and then subsequently heated up, in fact, to a rather high, uncomfortable temperature, and looked at the cortical areas, the cortical correlates associated with this thermoregulatory experience. With the PET scanner at that time, we couldn't really look in the detail in the nuclei. We were really constrained to look at the cerebrum, and you can see here a lot of areas associated with the sensory processes to do with thermosensation. And so again, it was a very novel piece of work, and fortuitous that being in San Antonio with the Apollo program just down the road in Houston, they're able to access this spacesuit. The subsequent work here in Melbourne is summarised in this slide, where individuals were put in an MRI scanner and then could be more frequently, in fact in this case twice over, manipulated with cooling the skin and then reheating the skin. You see here a bold signal measure taken at the time, and then you see the two physiological measures, the temperature of the core and the skin temperatures. And you see on the right here that the work was inspired by the earlier animal work done by Robin and colleagues, where they thought they could look in a specific area in the medulla, in the raphe nucleus, to be able to identify whether or not it was responding to these processes in humans. And so we took a region of interest approach through the medulla, as well as specifically in the area we could identify as the raphe, and that was then shown to have a specifically higher increase in this state in the cooling versus the non-cooling states. And so it was a first, I think, highly, if you like, impactful way of translating knowledge from the preclinical work in the rodent model across into human studies, guided by a specific region of interest to drive an understanding of where these nuclei were down in the human medulla. Subsequent to that, we moved on to look at some other interactions, as I mentioned, between thirst and, in this case, pain. Michael Farrell led this work. And the question here was, what would happen to the brain areas that we could see activated during the thirst experience when they're also given a pain phenomena, a thumbscrew, as delivered by Michael. And he's very good at that. If you know Michael, he's done that to quite a few people. And it's also something which I guess our volunteers for these experiments had to be comfortable with, that this was a fairly demanding experimental paradigm, generally speaking, from these physiological experiments to be involved in with us. And it was, I think, particularly challenging in this case with people having an infusion, then having that rather uncomfortable experience, but then further pain induced to see what would happen in the brain. And what we could see is that there was modulation of these responses, and in fact it was an enhancement by the joint experience there of both pain and being thirsty. So the role of modulating the thirst, we couldn't extinguish it. One of the questions was, would we see that thirst activation just extinguished by purpose of the pain stimulus? But it did not unreasonably, I guess. What we saw was an enhancement in the activity of the areas, particularly where there was overlap. After that, and again, this was part of the fantastic journey with Dick, you may remember that there was a French heatwave back in 2003 with a very large number of deaths, over 70,000, I believe. And so we had been studying thirst, and we'd been studying getting people to drink in scanners. Rather difficult, in fact, to get people to drink while they're lying prone. We had to use very small tubing and other ways to get people to either drink or to sip or to just wet their mouth and expectorate the liquids. But we had that expertise, and so we thought, why are these elderly people, what are the brain processes involved in elderly people being unable to fully rehydrate during these periods of extreme heat, hot weather? And so this experiment was done in two groups, a young and an elderly group. And you can see here the maps of maximum thirst. You can see a lot more activity in the anterior cingulate. And you can see after drinking, both groups have pretty much extinguished that activity in the brain. But when you look over here on the far right, what we identified, and this was very much Michael Farrell's work, was that the amount, because what we also measured was that the amount of water drunk, as shown down here on the bottom left, in the elderly group was significantly less than what the amount of water drunk by the normal or younger age people. And we could correlate this and show that the diminution in activation within that brain area, this is the cerebral blood flow change or reduction, was a lot greater in people, the elderly people, for a proportionately less amount of water drunk. And so it really came down to this, if you like, process, the mechanism by which that area of the cingulate was being, if you like, diminished in blood flow as a consequence of a small amount of water intake as compared to younger people. And so again, an important conclusion of that is not just relying on one's own, in the elderly group, one's own sense of requirement to rehydrate, but to actually look at approaching a more scheduled approach to drinking would help to avert future situations. I mentioned we did a slight excursion. We were still exploring this question of satiation, and the question of sexual appetite and satiation came up in conversation. I can't quite remember where it was, but it was an interesting thought. And then it turned out there was a group, in fact in Groningen in the Netherlands, that were doing some experiments looking at human sexual behaviours in the scanner. And so, as you may imagine, Dick reached out and made a collaboration. Michael Farrell and I were not really sure about this at all, but we kind of went along for the ride, you might say. But when we got there, it was obviously a very rigorous scientific facility, and we were able to do an experiment, probably the only place in the world you could do it at that time, which was to look at penile stimulation by these males' partners, and then measure their levels of arousal and then ultimately orgasm lying in the scanner. I think the most challenging part of this experiment was actually having the language between us that we could use to discuss the experiment and the results when there were other people also in the room, so that it wasn't, immediately embarrassing in fact how we were manipulating people and getting them to do things in a scanner. But the key point really was, and you see here on the bottom left, this, if you like, time profile of different individuals, times of tumescence and then ultimately orgasm and post-orgasm. And it was again that question, how can we identify those brain areas very specific to that experience of sexual appetite, somewhat diminished, remanifest and then satiated. And so we found a really interesting network of brain areas from that work. And it was, I guess, an excursion to one side which we didn't follow on with, but it would have always generated some interesting new experimental ideas. So then just quickly to finish up, there was a lovely experiment done, again here in Melbourne, looking at connectivity of the brain, the ascending pathways from the areas known down in the lamina terminalis, in the initiation of thirst, where they're sensing the chemistry, the blood chemistry changes in osmolality and sodium concentration. And so it revealed interesting areas in the midbrain here. You can see in some of the subcortical nuclei, and then also in the lateral hippocampus. And again, some of the areas in the anterior cingulate areas came up, not the same that we saw in earlier experiments, so it was a different result. Some of the posterior cingulate that came up, again, quite similar, but not really, we think, associated with the experience or the consciousness of thirst. And so an exploration into these areas that are actually, as we know, wired into those deep brain structures. And as I mentioned, a series of other papers which really were very much to Dick's encouragement and I think you would say a really lovely way of just fostering creativity discussion, ideas, and then risk, and then adventure to look at what we could or couldn't actually identify. Now, one thing I mentioned was this idea of the components of the thirst experience, and we've had a paper prepared on this for some time. I just realised it's never been published, so we might look to finalise that and submit that. I hope Mike Farrell's got some time to work with me on that. So in conclusion, I would really love to just acknowledge the great debt I have to Professor Denton. It was a terrific time working with him over that period. His contribution in brain imaging, I think, is internationally recognised. He really invented the neuroimaging of interoception. We collectively published over 20 papers in that period, and fortunately 15 were in PNAS. And we had, I think, the first to really use neuroimaging to look at this origins of consciousness. Not necessarily consciousness per se, there have been other groups who have done that in the late 90s, but looking at the origins of consciousness. My experience was, Dick, that he never gave up. We never gave up on any paper being submitted, being reviewed on an experiment that didn't work first time, and he obviously never slowed down. Our last paper was in 2020, and it was really a lovely experience working with him. Just one final comment is from after Dick's passing, he contacted me, and I just thought it'd be worth reading his comments that the loss of our long-time mentor and our mutual collaborator, Derek, he was a great scientist, a charming conversationalist, a gracious host, a discriminating connoisseur of wines, and most importantly, a dear friend. Thank you very much.
So Gary, thanks for sharing that with us. It's a real tour de force that you've provided us with, and actually indicating work in progress. So it may be that Dick's CV is yet to be added to in terms of the work in preparation. And I think what you've really demonstrated as well is that Dick was visionary in many areas, and this area which you obviously have played a seminal role in as well, and how he could think big and apply new technologies in novel ways to solve left field problems which others hadn't been thinking about, and that's a very clear demonstration of that. I'd also like to pay tribute to both you and Graham Jackson, because the legacy of these experiments has been our very deep tour de force in relation to imaging, both at the Florey, the University of Melbourne, and also Victoria in general. Both Gary and Graham have been seminal in establishing the Victorian brain imaging capability, or biomedical imaging capability, should I say, and also imaging, advanced imaging here in Parkville and also at the Austin. And Gary in particular was seminal in getting the 7T ultra-high field strength magnet just next door. We also have a PET CT there, which is for research only, and 2 dedicated 3T magnets at the Austin campus. And this reflects the evolution, basically, from fundamental science, which continues, and there's been great work which has been done elsewhere within the Florey in relation to diffusion-weighted imaging and establishing perspectives about functional neural networks, but now extending into application in terms of looking at epileptogenic foci, establishing where they are in the brain, in terms of understanding the pathogenesis of Alzheimer's disease, work which Colin Masters and Chris Rowe have championed. And in addition, most recently, the acquisition of a HIFU, high-field ultrasonography, to actually now have targeted interventions. You don't need neurosurgery, basically, to provide very targeted lesions for benefit in areas such as the central tremor and movement disorders, for example. So really a long and exalted journey, and thank you, Gary, for your contribution today. Now, our final scientific presentation is from Professor Robin McCallum. Dick recruited Robin to the Florey in 1988, I believe, and Robin and his colleagues have since made numerous contributions to knowledge of how the brain via the autonomic nervous system regulates basic functions that maintain health. In essence, Robin's research has helped to define specific nuclei within the brainstem where these various autonomic functions are controlled. He delivers a talk today entitled Good Housekeeping, How Brain and Nerves Look After Your Body. Thank you, Robin.
Well, thank you very much, Trevor, and thank you for giving me the opportunity to speak here. Dick changed my life. He invited me to join the Florey 35 years ago, and I've collaborated with all of the previous speakers, and it's been a magnificent experience. So I hope you forgive the light-hearted title, but it's meant to say we're talking about physiology of the body, much less about functions in the brain, but how the brain looks after the body. So here's Dick. I should tell you, when he invited me to join the Florey, and I was in England in my lab, he rang me up about a month before I was due to come out and said, how are things going? I said, they're going fine, but the visas are taking some time. I hope they'll be ready in time. And the very next day, the head of the Australian High Commission in London rang me up and said, don't worry. There's a man with clout. When I came to the Florey, he really wanted me to work on things like this, like salt appetite in sheep. But to his great credit, he gave me the freedom to pursue my own particular interests and to interact with colleagues as I saw fit. I work on the autonomic nervous system, I've done for many years, particularly on the sympathetic nervous system, which you will have heard about perhaps in this context. This is the popular view, saying that it's a system of nerves which switch on in times of emergency, and they all switch on together. Well, 99% of the time it's not like that. Sympathetic nerves are highly differentiated to fit their functions. They innervate almost every tissue in the body, and they operate to keep those areas of the body functioning. They come in many types. They may look similar when you just look at gross anatomy, but they're functionally distinct. Here's just an example, a simple example from an experiment done by Doug White. And this is showing simultaneous recordings of the activity of sympathetic vasoconstrictor nerves going to two different tissues, going to muscle and going to skin. And this is when the rat, the anesthetized rat, was heated. And you can see that as it gets hotter, the skin activity goes down to zero, and this means that the blood vessels in the skin dilate to help lose heat, and those in the muscle actually constrict, and this causes a shift of blood to the heat-losing areas. They can't be controlled by the same system. So another thing, this is from my colleague Colin Anderson in anatomy, and these just show the different types of sympathetic nerve identified in rats. They're going to different tissues, and the nerves going to different tissues have got a different histochemistry. They know what they are. They know where they're going. So how many functional classes of sympathetic nerve are there? Well, as an exercise, I sat down to write what I thought, and I came up with a list of about 30 or so. And so this is the working hypothesis. This is what's propelled me forwards and tried and suggested many more things. So the working hypothesis is that each sympathetic class is driven by its own dedicated circuitry in the central nervous system. This may not be 100% true, but it hasn't been proven wrong so far. So here's the idea. You've got a hierarchy of sympathetic control. You've got the target tissue here. You've got the postganglionic neuron with its cell body in the sympathetic ganglion. You've got the preganglionic neuron in the spinal cord. And then there's various pathways from the brain, and we call these the premotor neurons. Now, this is the same list as before. The ones I've read out are those that we really don't know where the premotor neurons are. So I'm just emphasizing the point that there's a lot to be discovered. And I'm just going to take you on a little journey of what my colleagues and I have been doing recently to look at one of these. And that is what we call immunomodulator neurons.
Now, it's not surprising that the two super systems of the body, the nervous system and the immune system, talk to each other, but really not a great deal. It's only in the last few years that we're beginning to discover more about this. This is just to say this has been supported by government agencies, and these are the key people involved. So the one who really got this started when he was postdoc in my lab was Davide Martelli from Italy. Michael McKinley has been a key colleague for all of this work, and we're working together now in the lab. Song Yao has joined us also for quite a lot of this. Melinda Goga is critical because she does the assays and the brain analysis for us, and we've been joined more recently by Pedro Trevisan-Bau from Brazil. So here's my non-specialist view of what's happening with inflammation. When the body is damaged or invaded by microbes, the first thing that happens is you get a process of inflammation. Now, inflammation is an essential part of the body's response, but it has to be at the right level, because if the inflammatory process gets too intense and spills over into the rest of the circulation, this can cause severe problems. It can damage host tissues, cause shock and even death, as Clive described in sepsis. On the other hand, if the level of inflammation is too little, you don't get an adequate defense immobilized by the body, and the bugs can win. So it's quite important to get the level of host defense inflammation at the right level. So to protect against these systemic effects of inflammation, which may damage us, the brain has evolved some mechanisms. One of them is use of the hypothalamic pituitary axis and release of glucocorticoids. But the other one I'm going to talk about is sympathetic nerves. So this is the idea. Inflammation gives infection or injury. These are detected by certain cells, and these produce chemical messengers called cytokines, and those cytokines also activate other processes in other cells to release more cytokines. And this is a cascade, and the key cytokine involved in this, which is both a necessary and sufficient driver of inflammation, is a cytokine called tumor necrosis factor, TNF. So here's the idea of the inflammatory reflex, which I'm going to be talking about. So an immune challenge is detected by immune cells, and this produces inflammatory cytokines like TNF and produces inflammation. But the brain gets to hear about it, and the brain gets to hear about it, and when the brain hears about it, it initiates a negative feedback control which damps down the inflammatory process and keeps it under control at a reasonable level. So, how do we measure it? Well, we do this mostly in anesthetized rats, but we've done it also in mice and sheep. And what we do is we give a standard inflammatory stimulus. In this case, we use lipopolysaccharide, we use a medium dose, and lipopolysaccharide is a product of the cell walls of E. coli bacteria, and it works very well in setting up inflammation, and it's a standard model used by others. And then what we do is we cut or sham cut the nerves which we think are important and measure the effect on what happens in the plasma TNF. Now, when we started on this, we were misled. And here's the prevailing view at the time. The prevailing view at the time, this is actually, I think, a picture from 2017, but it was the prevailing view in the early 2000s. And it was popularized by a man called Kevin Tracy, who publishes in Nature, Science, and this is an article from Scientific American. And his view, which is strongly held, is that the vagus nerve is really important in this. So in this model here, the inflammatory stimulus is detected by the vagus nerve and transmitted to the brain stem here, and this activates the vagus nerve coming out, and the vagus nerve finds some way indirectly to talk to the spleen where this interaction takes place. That's the story. Well, we tested this. This is a measure of the inflammatory response to lipopolysaccharide in an anaesthetized rat. This is when, this is the control experiment, this is with no LPS, this is with LPS. We get a measurable level of TNF out of it, and when we cut the vagi, it didn't make any difference. But when we cut the other nerves that we thought were actually going to the spleen, that's the splanchnic nerves, there's a big difference. And cutting the vagus on top made no difference. So we think that he's got the wrong nerves, and this difference here, between here and here, is the amount that the inflammatory reflex, these sympathetic nerves, are holding down the inflammatory process. Right. So this pathway, we don't think it's the vagus, we think it's the splanchnic nerves, and this pathway mediates the inflammatory reflex, and it's powerful. So what about the pathway? This is the one that we think is happening. We think the negative feedback is through the splanchnic nerves. So is this inflammatory action transient or sustained? Is it just something which we measure at 90 minutes after we've given our stimulus? And does it affect other inflammatory cytokines? And the answer is yes, it does. So what we've got here are 4 cytokines that we measure against time, and these were 6-hour experiments. So this is what I've shown you before, that when the reflex is active, this is the direction shown by the arrow, it reduces the effect of TNF, and this is an effect which peaks at an hour and a half. If we look at other inflammatory cytokines, interleukin-6 or interferon gamma, their difference that it makes is a little slower to be obvious, but after 3 or 4 hours, there's a very clear difference. And we believe that the TNF is actually driving these secondary responses. But the exception here is interleukin-10, which is an anti-inflammatory cytokine, which pushes things in the opposite direction. And actually, the reflex enhances those levels. So it's a double action. It's suppressing inflammatory cytokines and enhancing anti-inflammatory cytokines. So where's it working? The splanchnic nerves go pretty much everywhere in the abdomen. The assumption was that it worked on the spleen, and it does amongst other places, but a series of experiments which I'm not going to describe to you, we've found that the action is actually distributed through a number of organs in the abdomen. And this is our latest take on it. This is from a paper published last year. Our idea is that the inflammatory signals talk to the central nervous system, and these are the sympathetic pathways out. And it seems that actually a different sympathetic pathway is suppressing TNF by working probably on several abdominal organs. And the ones which go to the adrenal medulla and release adrenaline into the circulation are probably the ones which are driving the increase in interleukin-10. So it's a double-pronged reflex. Well, I'm not going to go into any detail of that, but what about its central pathways? And this is, again, where we come in. This is what we do. This is where we're interested in the central pathways which are driving it. And these are experiments in progress at the moment. We're chasing it down the brain. Here is, again, the difference between cutting the splanchnic nerves and not cutting the splanchnic nerves. This is a measure of the reflex in the intact state. So that's the difference which the reflex is holding down TNF with an intact brainstem, with an intact brain. If we cut here, through the brain, behind the hypothalamus, but leaving the brainstem intact, same thing. It's not affected, so it doesn't need the hypothalamus. If, however, we cut between the brain and the spinal cord, the reflex disappears. So it's something in the brainstem, and we've been trying to refine it a bit since then, and this is our latest view of where it is. It's somewhere in the medulla, and this is work in progress. Final thing I'd like to say is this just an artifact that we're finding in our experimental animals, or does it matter? And if so, when? Well, I'm very glad that Clive spoke about sepsis earlier, because this is a key area where we think it may be important. I don't need to go into those details. But this I will share with you. This is a diagram put out by Richard Hotchkiss, a few years ago now, and this is describing the processes that are going on in a septic patient. In the early stages, there's a very strong inflammatory response, which has been called the cytokine storm. And early attempts to improve the outcome of sepsis thought all we've got to do is fight this cytokine storm and give antagonists for this and things will be better. Actually, people did worse. But in the later stages, there's a time of essential immune suppression. And our hypothesis was that maybe the inflammatory reflex is active, and it probably helps moderate the earlier cytokine storm, but maybe it persists to drive the later immunosuppression. So we did some experiments with Clive and Yogesh, again on sheep, and in half of the sheep, we cut the splanchnic nerves to disable the reflex, and gave them live E. coli from a septic patient, and watched what happened. And as we predicted, the ones with the cut splanchnic nerves, so without the reflex, had a stronger inflammatory response, higher levels of tumor necrosis factor, higher levels of interleukin-6, but lower levels of interleukin-10, the anti-inflammatory one, exactly as we would have predicted. But this we didn't predict. This is measuring the counts of the bacteria in the blood. Disabling the reflex seems to have enabled the animals to just throw off the infection in a very short time. And they got better quicker. So this convinces us that the inflammatory reflex is functionally important, but of course it doesn't always work in our best interest, and that's probably because evolution designed it for a different purpose. I think that we're still in the early stages of a long voyage. I just wanted to leave you with some thoughts that this was the inscription written on Sir Christopher Wren's tomb in St. Paul's Cathedral, which he designed and had built. Si monumentum requiris circumspici, which is Latin for, if you want to look for my monument, look around you. Same applies to Dick, I think. Thank you very much.
What a fantastic and apt final slide. I think it really said it all. And what you've really delivered to us, Robin, is a perspective of actually erudite science and sticking to your guns. You know, the importance of setting the record straight, you know, not the Vegas. You know, sympathetic nerve is important. But it also really sends a message of importance to the Institute, I think, and the importance of interdisciplinarity, multidisciplinarity, should we say, and that we need to commit to the area of neuroinflammation. It's multifaceted. It's not just, you know, my area in relation to multiple sclerosis. It's the perspective of the importance of the nervous system in areas such as infection. It extends to the role of inflammation in neurodegenerative diseases. And it also extends to that human environmental interface in terms of the microbiome. And I'm sure Jim's favourite cells, mate cells, you know, are implicated there somewhere as well. So as you said in that final slide and in the penultimate slide as well, I don't know which boat was it, but anyway, we are still in you know, sort of medieval times in terms of understanding some of these core issues. Much work to be done, and I think we're up for it, is the bottom line. Now, before I close, just one mea culpa. One really important person I didn't acknowledge earlier was Mike McKinley, who was absolutely similarly involved in organising today's scientific program. So thank you very much, Mike. And thank you to all the speakers today who've contributed really what I think we would agree were fascinating talks, and really talks which have helped to honour Dick's legacy. And I'm sure that Dick would have been thrilled to know how your important research that he cherished has progressed, and the ongoing passion you have for your science, and ultimately its application to enhancing the health of our community. So well done to all our speakers. Thank you very much. Now, I've absolutely failed as your MC today, and we're running behind time, but obviously we do have to have a short break, and I'd suggest that people come back into the auditorium for the second session around 1:10 p.m. Thanks very much.
I'll just ask everybody to take their seats and we'll get started with the next part of the program. So I wonder if you can dim the lights. Is anyone on light duty? Can get people to then put them up again, dim them and put them up again. I'm fine, I'm just trying to get people to be quiet. Okay everyone, we're going to get started now. Well, just to get them to be quiet. Welcome, rabble, welcome everybody. Attention in the auditorium. Okay, I know that you've had a morning of sitting. I could get you to do the coconut thing that the physios make us do to limber up, but that might take a bit too long. So thank you for resuming your seats. I'm Professor Jane Gunn, so I'm Dean of the Faculty of Medicine, Dentistry and Health Sciences here at the University of Melbourne. I'm also on the Florey board as part of that role, so it's great to be here this morning. I'd like to start by acknowledging the traditional owners of the lands upon which we're meeting and having this oration, the Wurundjeri people of the Kulin Nation, and pay respects to elders past, present and emerging, and particularly any Indigenous colleagues here in the auditorium today. I also want to make note that one of our living legends, Professor Marcia Langton, had a very prominent place yesterday in the media with the finalisation of the Voice to Parliament and the referendum wording for later in the year. So I want to thank you all for joining us this afternoon. We are running a little bit behind time, but I'm sure you'll find it worthwhile that you remain with us, whether you're here in person, whether you're online, as we honour the life and work of the Florey's founding director, Professor Emeritus Derek Denton, AC, fondly known as Dick. And I must say I was most privileged to have met Professor Denton a number of years ago when I had the great honour of going and talking with him about how we might shape the Derek Denton oration, which is the thing that you're going to be here for today, the oration in science and the arts. And this began as, I think it was about 3:30 in the afternoon when I went to have the meeting, and Dick was in his office and he was really happy to see myself there, and I went with one of our advancement team members, and he had a couple of champagne flutes on the table and walked in, and I said, "Oh, you're having a celebration?" He said, "Oh no, well I'm having a visit from you." And so I thought you might need a little bubbly for the afternoon. And I was like, "Oh, okay. Well, how could you say no?" And then what was going to be a 20-minute visit, I think, turned in. I was there about an hour and a half, 2 hours, and I was really treated to the life and times of Derek Denton according to Derek Denton, which was something I must say I will never forget. And then, of course, had visits with him from that time on. But always learnt something, and it's just great to be here today and to be invited to MC this final part of the program. So I'd like to also make note that this year's Derek Denton Oration in Science and the Arts is going to be delivered by Dr Robin Williams, who has been here for the first part of the proceedings today as well. And I know that I think with my life of Derek Denton according to Dick, Robin Williams' name was mentioned within about the first 5 minutes. And it was basically that, should anything need to be worked out about this oration in times to come, because who knows how long he was going to be there, and he'd only just met me, and who knows what was going to happen to me. The go-to person for any further information was Robin Williams. So I do remember that. Dr Williams is a science journalist, broadcaster, presenter of Radio National Science Show and Occam's Razor, two of my most favourite programs, deputy chair of the Australian Science Media Centre, and great friend of Professor Denton and his wife, Dame Margaret Scott, and has been a friend for 50 years plus. Before the oration, though, you're going to have to hold your attention, because Professor Trevor Kilpatrick, head of the Florey Department at the University of Melbourne, as well as director of the Florey, as well as Professor Denton's son, Angus, is going to speak with you. But first, we have a very special treat, and that is that we're going to show a video, and I'm going to have some assistance here to get that going. And this video has been put together by members of the Florey as their messages and tributes to Professor Denton.
Hello from Sweden.
My supervisor, Ben Anderson, met Derek in Cambridge in 1952-53, and they've been close friends and collaborators since then. They had a lot of mutual contact between the department in Stockholm and the Florey Institute. And I was there on a postdoc in 1984-85. And Derek was always a strong supporter of young scientists. During my stay, I worked mainly with Dr. Michael and Kim. Some years later, Dick organized a conference, a Swedish Australian symposium, and in connection with that, went out to his place in Gooding National Park, north of Sydney. And that's one of my strongest memories of Dick, because on the way out, we bought a bucket full of oysters, and I've never eaten so many oysters.
Since I came to Florey as a student 52 years ago, and he was my PhD supervisor, and then I worked alongside him. He helped me get away to a postdoc at the Karolinska Institute, but I came back to Florey after that, and we worked together until virtually after they died. So he was very keen on curiosity-driven research, blue sky research, and for ideas that might come out of the blue, and people had an interesting passion for the way you should investigate them.
Derek and I laid out an answer how addictions came about, what deep-rooted brain mechanisms they latch on, and why addictions might be referred to as artificial instincts. Our new concept fundamentally changes how science is translated into medicine. Rest in peace, Derek. You will be missed, you will be remembered.
In truth, I knew Dick only for occasional scientific meetings. But from those brief encounters, my memories are of a great intellect, educator, and human being. Indeed, one of my prized possessions from those meetings is a signed copy of The Hunger for Salt. At my age, one might be indulged to look back and ask, who are the scientists that have made the greatest contribution to this field? My vote, unequivocally, goes to Derek Denton.
I'm Linnea. Max is my dad, and I knew Dick, thanks to my dad, when we lived in Australia, 84-85. But I also know Dick from several years later when I came back to Australia on my own when I was 18. Dick then introduced me to his son Matthew and Matthew's wife Annie. They ended up taking care of Josh, or helping out to take care of Josh, which in the end meant that I actually moved in with Annie and Matt, and there started just over 6 months of very warm memories together with the family. And I think my strongest image of Dick is probably also from Booty, going out there with him, him in his silly sun hat on his terrace, eating oysters or talking about oysters, with a chilled glass of Chardonnay in his hand. And that was typical Dick, I think.
I moved over to the Florey 20 years ago, and I've known Dick ever since then. Our relationship started as a collaboration, looking at similarities in the mechanisms between salt appetite and addictive behaviour, and through that collaboration we became close friends. I think I would equate Dick to being a terrier. He was the kind of person that would just not let go. He was determined and enthusiastic and ambitious, but collaborative as well. So he knew the strength of teamwork. He knew the strength of developing teams with complementary skill sets. But that terrier instinct was, I think, what saw him through to the levels that he achieved. Dick was engaging, personable, always very interested in anything you had to say. And he was very well read, so you could have a conversation with him about many different things. But he was not judgmental. He was very much, he takes you as you are. He was also very good at caring for people. He was very, very thoughtful and considerate of people and their needs, and a real gentleman.
So it's wonderful to hear those tributes. And now I ask Professor Trevor Kilpatrick to say a few words.
Thank you, Professor Gunn. Derek Denton was a visionary, a man who not only had big ideas, but who also combined his vision with an unbridled tenacity to make things happen and to make a real difference. There were many examples of that, most significantly enunciated through his scientific contributions, his love of the arts, and most substantively, the Florey. Now, the history of the Florey and of Derek's seminal contribution to our history as our founding director is known to us all. That history, of course, started with the Howard Florey Laboratories in 1962, dedicated on the 30th of August 1963 by the then Prime Minister Sir Robert Menzies in the presence of Sir Howard Florey, President of the Royal Society. And what is there to learn even today from Derek's leadership in those formative years? Firstly, he made sure that all involved, including government, major donors such as the Potter and Meyer families, and the scientific community, appreciated the Florey was important and that it had a key strategic focus, commencing with work on salt and water metabolism, essential to health and well-being. Secondly, he engineered that the Florey was independent, but with strong and enduring partnerships with key stakeholders, not least the University of Melbourne. And thirdly, he and his supporters ensured that the name of Howard Florey not only be used to capture attention, both locally and internationally, but that it also be used to emphasise a continuing commitment to a translational focus. And it is to Derek's credit that these principles became core values that remain strong at the Florey to the current day. That is focus, strong stakeholder engagement, translational ambition, and striving for community relevance and impact. Now, Derek retired as director in the 1990s, but he remained integrally engaged as a research scientist, as you've seen in the talks today. Granted, the scientific focus has evolved. That is, the interrogation of salt balance expanded to the study of neuroendocrinology and of interoception. That is, the unconscious control of key body functions that are orchestrated by the brainstem and hypothalamus. And in the early 2000s, there were key amalgamations and a new capital works program and a commitment to a focus on neuroscience and mental health. Now, the emergent focus on neuroscience was in fact a natural evolution of the flourishing work of brain physiology within the Florey, as we've heard today. It also reflected community-based recognition of the importance of understanding key processes such as learning and memory, and identifying new treatments for important neurological and mental health-related disorders. Despite his advancing age, Derek continued to adapt, and this was reflected in his science. For example, embracing the power of magnetic resonance imaging, as Gary Egan has indicated today, and its key role in unravelling structural and functional neuroanatomy, which remains a key focus of the Florey to the current day. But on a lighter note, one technological advance that Derek did not embrace was the computer. Now, Matt and Angus may have an explanation for that. I don't actually. And superficially, I find it an outliner because Derek was certainly no Luddite. And perversely, as someone who receives about 250 emails a day, I'm actually envious of Derek's rejection of the computer. And perhaps paradoxically, it was one of Derek's best insights, explaining his unbridled and enduring capacity for deep thought, uninterrupted by the capricious sirens of the virtual world. And despite his aversion for the computer, Derek interacted very productively with each of his successors, either face-to-face or by phone. And he was certainly very proficient in mobile phone technology, to which many of his friends and colleagues, including those in this room, can certainly attest. And here on reflection, with some poignancy, I can certainly speak to Derek's generosity of spirit. I well remember a knock on my office door around this time last year to find Derek outside, having decided that a brief jaunt in his car from Toorak to Parkville in Friday afternoon traffic at the age of 97 was a good idea. But he thought it important. And in essence, he wanted to congratulate me on my new appointment, to provide his unique perspective of the Florey's proud history, and to make sure that I understood I had a significant responsibility. The baton had been handed on to me. Don't drop it. And importantly, he was also there to signal his support for the key decisions we were about to make concerning our emergent strategic vision for a future Florey. On a personal level, I appreciated his gregarious but also tenacious spirit. Perhaps that exemplifies his capacity to deal with Melbourne traffic at the age of 97. And as a fellow scientist, I admired his principal raison d'etre, that is, his inquisitive mind, his erudite dissection of key issues, the way he promoted curiosity in himself and others, how he brought people together, and how he remained unwaveringly committed to excellence in academic pursuits. That is why Derek, or Dick, inspired me, and why I think he continues to inspire Florey scientists to the current day. Dick's generosity of spirit was also obvious, as exemplified by his donations and of his precious manuscripts and artwork to the Florey. And that generosity was apparent right to the end. 3 weeks before his death, perhaps similar to your encounter, Jane, Dick and I were sharing a fine wine in his home, discussing science and the meaning of life. Those exchanges with Dick will always be cherished, and it's something which I will sorely miss. And what did he think about our emerging plans for the Florey? In particular, our focus on finding treatments for dementia, curing epileptic syndromes, preventing and repairing damage to the brain, and our strive to understand the molecular drivers of psychiatric disease. In fact, he was widely enthusiastic about those plans, and for the same reasons he championed the concept of the Florey 60-odd years before. In essence, a strategic vision, driven by in-house expertise, strengthened by the right partnerships, and with a commitment to make a real difference. And the infrastructure we will use to achieve those goals will be near and dear to Dick's heart. Neuroimaging, the sheep facility, vital for critical care research we are conducting as indicated by Lindsea and Clive today, and for advances in med tech pertinent to the neurosciences, and a continuing focus on neurotherapeutics. And it is with that shared focus and collective strength that we continue to strive to build a Florey of which Derek would be proud. Val Derek Denton, a great scientist and polymath, your legacy burns bright with us.
Thank you, Trevor. It reminded me with the car ride on the Friday night of the car parking space where the Emeritus professors park their cars off Haymarket Lane there. Not used that much throughout the pandemic, but prior to that, Derek had his spot and everyone else had their spot, and there were a lot of vacant spots in between just to make sure that he could get in and get out without too much trouble. And that was, I don't know how, it was a pretty poorly kept secret. I'm now delighted to welcome to the podium the son of Dick Denton, Angus Denton. Over to you, Angus.
I spent a lot of time last night furiously typing away, and I think I'm going to throw away 3/4 of my notes because a lot of what I thought I'd cover has been covered. I want to pick up on a few things. Professor Gunn. Derek, according to Derek. I think everybody in the room has had a discussion with Dick talking about what he's done and how he's gone about doing it. So your comment really did make me laugh. I think the lingua franca on the slides from Brian Oldfield and the word wonder, I think that's an extraordinary word. I mean, just a word in inverted commas that I could label my father about his view of life and the world as wonder. I think it's fantastic. So I don't know what the collective IQ is in this room, but for me to be standing here in front of you, addressing you, it's actually scary. But I'm actually really humbled, and I think I can speak for Matt as well. I'm just so humbled for the respect for Dick. It's just, yes, I can't find any other words. So there's an aphorism that if you work at something you love, you'll never work a day in your life. By this standard, my father never had a job. This institute and where we meet was an all-encompassing passion for Derek Ashworth Denton, a lad from Launceston. The reality is that we knew little about Dick's family, his side of the family, but his father was a motor bodybuilder and not an academic, nor was he medical. However, at Launceston Grammar he studied chemistry, and maybe that was the beginning. He chose medicine as his pathway, and to take that path in Tasmania you basically had to leave. So leaving Launceston in the 1930s for medical school at Melbourne University, armed with a scholarship to Trinity College, must have been a mind-expanding experience. It's quite a shock to reflect and realise that when Dick was a medical student, there was no such thing as antibiotics. The prick of a rose or the nick while shaving or a person's cough on a bus is potentially fatal. I can imagine that medicos of the time would feel extremely frustrated and stymied knowing that seemingly healthy people could just slip away, or that evidently successful surgery was defeated by post-operative infection which they were powerless to control. And I suspect that that's where the hunger and the dedication in Dick Denton came from, to know how to better save lives and understand how something worked or actually didn't. So the suggested topic for me today was to explore Dick's love of science. Dick was a doctor with a curious mind, and science was a tool. To say Dick loved science, I think is a bit misleading. I think he loved ideas and he could see possibilities. Science was his tool to realise those possibilities, a bridge between an idea and a solution. But what is science? The Oxford English Dictionary describes it as the systematic study of the structure and behaviour of the physical and natural world through observation, experimentation, and the testing of theories against evidence obtained. It sounds like a verb. For the individual scientist, I think we can say that there's a series of mental and physical disciplines that must be rationally applied. So it's clear Dick started with ideas, he decorated them with words like how and why and what if, and then moulded that into a theory, and then came the verb, to science, and a lot of mental and physical discipline was then applied. I think I might be giving you the wrong impression though about Dick. You see, it wasn't all about him. Dick was a gregarious man, to say the least. He loved ideas and he loved discussing them with people. And he was happiest listening to other people's ideas and confecting an environment for like-minded people to talk. In fact, it was baked into the brief given to the architect, Barry Patton, when designing the building adjacent to this one. You see, science is a team sport. We've heard about that today. To tackle the big ideas, you need more than one set of mental and physical disciplines. It's impossible for a single human being. So Dick's love of ideas shared, and his gregarious nature meant that he built teams, but the secret sauce was conversation. For those of you who joined Matt and myself at Ripponlea yesterday, you would know that conversation was a huge part of Dick's life. And we have heard this many times over today. It's all about the conversations people had with Dick. The original Florey building had various teams in their labs, and all the other spaces were common. In particular, there was the library, and there was the tea room. And there was a twice daily ritual of the tea with one of the largest teapots I've ever seen. Everybody mixed, from eminents such as Sir Roy Douglas, Pansy, Wright, to the animal technicians. Physiologists could talk to chemists or geneticists, and they might have an insight into each other's dilemmas, and frequently did, from what I understand. Because Dick loved ideas, they didn't always have to be scientific. The research he was undertaking was rooted in animal and human behaviour, honed through millennia of evolution. And there were many insights to be gained into the nature of the physiology that he studied, but recounted through literature, anthropology, poetry, and art. In rereading Tim Hewitt's book, The Sheep Hilton, you realise that Dick's work in the job that he didn't have was infinite. It could never end. For every advance they made, new questions arose. New avenues appeared, and exciting and surprising relationships presented themselves. For this was basic science, inquiry for its own sake. I cannot recall hearing the language of KPIs. I only heard the excitement wrought of ideas. Collaborators could find their own paths, be it at the Florey or other institutions, everything branching and growing, and a bit like a fractal, but without the continuous repetition. Today's presentations prove this. Could Dick ever have envisaged that his original work, his original ideas, would lead to maybe treatments for anorexia? So, the Florey has this towering reputation internationally in a way that the general public just does not understand. It has a roll call of extraordinary minds who have visited as fellows and contributed their ideas, and we cannot forget and that has been and still is a nursery for students to develop their own ideas and go off and follow their own stars and revealing great things and again generating more ideas. So I don't think science is actually a verb. I think it's a state of mind where ideas can flourish. So Trevor, let's go warm the scanners and let's go looking for the seat of where it lies. Let's get cracking. And finally, I'd really like to say, I feel that Dad's work and his legacy is in brilliant hands. Long may you reign. Thank you.
Thank you, Angus. So now we move on to the formal Derek Denton oration in science and the arts. This oration has been delivered first in 2010 and many times since that. It has, as you would know, Dick was very well connected and the orations have hosted high-profile international guests, including Lord Martin Rees, one of the world's preeminent cosmologists, among many others. In 2018, we received a gift from Dick Denton, which established a trust so that the future of the Derek Denton oration in science and the arts would not be left to chance. As I recounted earlier, there was a very clear view about what the oration what was the secret source of the oration. Well, one of the things was to get a good person to do the oration, get a brilliant mind to spark interest. And then after that, the most important thing of all was that people from different backgrounds, from different ways of thinking, at all life stages, but particularly getting younger people into the room, would then share conversation over a meal. Today we will do a bit of a bigger version of that, where we will have the oration and then we will share conversation over some bubbles and food. And I think that Dick Denton would be very pleased with that. And I think he would also be very, very, very pleased to think that since his passing, that the first time the oration was delivered, that it would be delivered by his long-time friend, Dr. Robin Williams. I'm absolutely sure of that. I have a very long CV here that I could read out about Robin, but I know that you, he's shaking his head, I know that you are well known to the audience and everyone would feel they know you very, very well. You are a national living treasure and that was proclaimed in 1987, so that's an absolute fact and we would all agree with that. And you've made many documentaries, books, and you've kept so many people well informed in science. And I'd like to invite you to the podium, Robin, to give the Derek Denton oration in science and the arts.
Thanks so much, Jane. In fact, a couple of my colleagues call me a national living fossil. I came out here some time ago and I always was so thrilled with the idea of Australia because I had friends there who turned out to be eminent. I didn't realise that at the time, and they had a stream of brilliant professors who were all so athletic and young and wonderful. I thought that's what Australia was like. I came out here just at the time Lucky Country was published. Well, for me, there's always been a kind of spectrum. of people in Australia when I actually got here. And it goes from Bazza Mackenzie to Barry Jones. And on the female side, from Pauline Hanson to our dear Denton friend, Robin Hughes, who was a colleague of mine and taught me to edit, and then became the Deputy Chancellor of the ANU. And I've always wondered whether this strange polarity in Australia would be solved by the future. In other words, would we become the gambling and coal capital of the world, or, as they used to say, the clever country? Barry Jones didn't like that phrase, the wise country, perhaps. Anyway, I've called this Two Worlds, One United Universe, the Denton universe, of course. I met Professor Derek Denton for the first time in 1972. I was in a cafe, or pub, at King's Cross in Sydney, not far from the ABC Science Unit's office at the top of William Street. The unit had been set up barely 8 years before, but already Derek had recorded a series of talks that became a book called Nervous Insights, which was remarkable for its wide-ranging analysis and predictions of investigation of this complex topic. Several ABC Science colleagues were gathered around this genial star of media and laboratory bench. He was smiling and directly engaging, and we seemed to become chums almost instantly, for that was one of Dick's unique attributes, the gift of friendship. Another was his obviously multicultural mind. The science was not stuck in a box. I was surprised I had not, on my landing in Australia, had to endure an artistic or intellectual wasteland, as some distant observers had feared. I knew how to connect. But it was remarkable how even famous Australian writers, such as Shirley Hazzard, who wrote Transit of Venus, were bleak in recalling their own supposedly sterile youth. She says, "In the circles where I was raised, I knew of no one knowledgeable in the visual arts, no one who regularly attended musical performances, and only 2 adults other than my teachers who spoke without embarrassment of poetry and literature, both of these being women. And as far as I recall, I never heard a man refer to a good or great book." Shirley Hazzard, not me. Yet he was Dick Denton, born in Launceston, Tasmania, who relished paintings, ballet, music, literature, the lot, but not computers, of course. And what's more, averred that it was the brain's function to integrate them all via consciousness and much else. Aren't all these elements of thought quite different? Aren't we condemned to those notorious 2 cultures? Apparently not. And here's why. A new book by Jonathan Kennedy says this, I quote, "The ingenuity that is evident in the Paleolithic European cave art plays a part in Homo sapiens' rise to world dominance. Our species are said to have triumphed over all others because they were uniquely capable of what paleoanthropologists call symbolic behaviour. In other words, we had the capacity to use language and art to express and exchange ideas. Ideas. The ability to think and behave in complex ways allowed us to plan, cooperate, and outcompete the bigger, stronger Neanderthals and other human beings who were less skilled." Jonathan Kennedy. But how, asked Denton, did this actually work in a brain with millions of neurons? So many chemical connections, and the hormones needed to play so many tunes conducted by the maestro called the pituitary. All in a body needing constant homeostasis, having left the sea and migrated onto the new home dry land, where every landscape had a different complement of essential minerals, not least salt. We discussed this quandary incessantly in various small broadcasts. But of course, as I said, Dick was an ideas man. Yes, he did do lab work to elucidate parts of brain and body function, but he wanted to take things further. His exploration of instinct in his book 1982, The Hunger for Salt, which you've heard about this morning, and tackle the big one, the pinnacle of life, he called it, in the Science Show series, and his book in 1993 on consciousness and self-awareness in humans and animals. And the book, of course, is replete with artistic flourishes, quoting Shakespeare, for example. What a piece of work is man! How noble in reason, how infinite in faculty, in form, in moving, how express and admirable, in action, how like an angel, in apprehension, how like a god, the beauty of the world, the paragon of animals. And all that with just grey matter. No, Hamlet was quoted with some irony because Dick loved animals and wrote lots about them. The point about the arts in his book was to demonstrate that they are the glue of civilization, not just the decoration. The arts are the glue of civilization, not just the decoration. And he used from Richard II, "My brain I'll prove the female to my soul the father, and these two beget a generation of still breeding thoughts." Male and female together, not separate. To understand this sort of dichotomy, for there are many in the discussion of consciousness, I must follow Dick Deedee's charming example and drop a couple of names. Dick was able to drop them because he was constantly in the company of heroes. Once, for example, long ago when I was visiting San Diego and running along the clifftop path overlooking the sea near La Jolla, I saw a familiar silhouette ahead. Two of them together, actually. But breathing hard, I addressed the one I knew and said, "Dr. Denton, do you have $25 for a cup of coffee for an old digger?" Dick reached into his pocket to find some cash, recognized me, and then gave a tremendous laugh. Then he introduced me to his companion, who, of course, was a Nobel Prize winner. Dick seemed to know everybody. But two Nobelists we both knew may illustrate the difference in thinking between the conventional thinkers, ones who just do facts, and those who do real ideas. I'm thinking of Watson and Crick, so different to each other, and only one of them close to Derek Denton. In the mid-1900s, I was a visiting fellow at Balliol College in Oxford, and it so happened that Dick was there at the same time. And so was Jim Watson. He often joined us at lunch, which was difficult because Watson had no interest in ourselves, but liked to assert various attitudes. This puzzled me, as his original studies had been of birds, and I wondered whether his interest had been of their bare numbers and nothing to do with their delightful behaviour, songs or plumage. Nerdy, in other words. Watson's disengagement from normal human interaction was illustrated one day when he gave a lecture to the postdocs at Balliol and spoke, or rather muttered, for an hour to his shoes. None of us had any memory of what he talked about, but he did have some appreciation of patterns. You will recall, however, when he stole Rosalind Franklin's photo of DNA. That's photograph 41. There was actually a play about it called that with Nicole Kidman in the West End of London. And he spotted the pattern of the double helix. But in normal discourse, he was nerdy. Francis Crick, however, was so different. He trained as a physicist. He did his research on sonar during the war. He was a friend of Rosalind Franklin's and indeed of Dick Denton's, and came from the USA while we were still in Oxford to speak at the Oxford Union. It was packed and the atmosphere was exciting, as if we were greeting a rock star. After his address, Derek nudged me and said, "Ask him the question." So I did. Dr. Crick, I was slightly nervous at the time, did consciousness evolve, or did it appear de novo as our brains became complex enough to produce it? Evolve was the firm, smiling answer, as Dick knew it would be. And he too gave a jolly smile and waved to Crick. The conversation continued when Crick was back in California. We'd be in the middle of some highfalutin speculation when Derek would remark, let's see what Francis thinks. And he'd just call him up, and the conversation would go on for ages. Being at Balliol, by the way, taught me lots about Dick's understanding of economics and of enology, another example of his expertise, as you know, of wine. Do ask the butler, he confided, to take you to the cellar because their collection is stunning, and ask for a couple of bottles of Chateauneuf de Pape. So I did. The first surprise was that the butler, a young woman, that's Balliol for you. The second surprise was the price. My 2 bottles of the Old Telegraph, Dick's favourite, in the shops would have been, say, $200 to $300 at least each. The college charge for the fellows was just over $15. Why? Because that was the price paid when the fellows in charge of wine had bought them in France years and years ago. So they just kept that price going, which I think is very civilised, and every bottle shop in the centre of Melbourne, Carlton, should do the same thing. That was the kind of flair that over the decades enabled Derek to raise so much research at the Florey and to finance the series of lectures on art and science and so on. And yet again, Francis Crick illustrates that connection with his real understanding of effective science communication beyond the ivory tower. It's gossip, he insisted. Gossip, not the pejorative term. Not a pejorative term, but again, the glue of civilization, as another Oxford Don, Robin Dunbar, showed in his book on the evolution of language. Gossip substituted for grooming, as supposedly we came down from the trees. If you just groomed yourself without looking up, some great big saber-tooth or whatever it was around would gobble you up quickly. So you had gossip and chat instead, and so language evolved. Talk was of group interactions and of ideas. Gossip about science for Crick and Denton is linking phenomena and ideas, the essence or originality, innovation and nature. Some of the greatest scientists in history have pioneered this and in a way been forgotten. Which brings me to the question, what about Dame Maggie Scott and the first person in history to be called a scientist? I know one person in this room, apart from me, does know. In 1834, the Cambridge Don William Whewell wrote a complimentary article about Mary Somerville, a Scottish researcher whose erudite books brought together previously disparate fields of mathematics, astronomy, geology, chemistry, and physics, so clearly that the texts became the backbone of Cambridge University's first science curriculum. He called Somerville a scientist in part because, quote, man of science seemed inappropriate for a woman, but more significantly because Somerville's work was interdisciplinary. She was no mere astronomer, physicist, or chemist, but a visionary thinker who articulated the connections among the various branches of inquiry. According to Somerville's biographer, Catherine Neely, Whewell's coinage of the word scientist was not meant to be merely a gender-neutral term. Whewell wanted a word that actively celebrated the peculiar illumination of the female mind, the ability to synthesize separate fields into a single discipline. The first scientist in the English language was a woman, and you got it right, Olympia. The first wife of Derek Denton and the last was a woman from South Africa, Maggie Scott, whose life as a dancer and choreographer, no, this is not a leap too far, was based on knowledge of the human body and how damage can be prevented and fixed. Her own recovery was several times thanks to modern medicine, and her last triumph on stage that I witnessed was at the Sydney Opera House when she starred in Nutcracker, written for her by Graham Murphy, one of her students way back, as she danced superbly. Having had an artificial hip put in, she was 69. Derek had, of course, been in touch with people he named Rupert and Kerry and their extended families. We knew who he meant. And the hall was crammed with the A-list. Maggie had founded the Australian Ballet School and run it for 40 years. Both, by the way, were AC, as you know. But Derek had long been involved directly in ballet and knew its workings and needs very well. It was not an add-on, but connected beautifully with his work as a scientist. I understood this because of a day in 1954 when I was in Budapest, aged 10. My parents took me to a full-scale performance of the Bolshoi Ballet starring Galina Ulanova. Now, I knew nothing about dance as I walked in. 3 hours later, I was in tears both of joy and of heartbreak, as the full force of great art had somehow taken over. How was this? I was 10. And don't forget, it was after World War II, and all the theatres had been closed for ages. I had no experience of this previous form of art. And so somehow, this stuff responded. And I discussed it with Maggie and Dick when staying at their beautiful house in the National Park, just beyond Woy Woy, where incidentally Spike Milligan used to listen to the science show with his mother. Maggie knew the dancing techniques. Derek knew the way we are primed to respond. We learn new forms of display and storytelling. David Attenborough has likened the songlines, for example, in Indigenous Australia over thousands of years to modern movies. Both song and dance have narratives and ways of reaching your imagination, both using similar methods really and having a similar sort of impact. Afterwards, I descended from the Denton house. They couldn't do that at the time because the knees were too sus. I went down the steep natural steps to where the forest meets and even touches the shores, and I went to swim, lying on my back, looking up at the sky. A sea eagle flew from the trees and hovered above me for what seemed like many minutes as I contemplated the blue water and deep forest and watched the bird. I knew what E.O. Wilson meant by biophilia and what Derek said in his book Pinnacle of Life about its significance. Yes, wonder. And as Professor Zecchi from the University College London remarked in a lecture here in this series, the artist is in a sense a neuroscientist, exploring the potentials and capacities of the brain, though with different tools. How such creations can arise, aesthetic experiences can only be fully understood in neural terms. Such an understanding is now well within our reach. Within our reach. That is why Francis Crick, thrilled by the quest, was working to within 2 hours of his death on that understanding. And yes, according to Christoph Koch, who was working with Crick at the time, he assured me that the paper was published, Consciousness Biophilia, published work 2 hours before Crick died. And that's also why when released from COVID restrictions, when I flew to Melbourne to see Dick, he brought out 2 bottles to celebrate. One a Bollinger, the other a superb Chardonnay decades old and still marvellous. And we recorded a last interview, which I broadcast soon afterwards. It was about the sneaky way addiction to some narcotic drugs is wired into the thirst part of the brain, the part you saw just now, from which it's very hard to remove. My final tale about Dick is on the Pinnacle of Life, the last book. I was at the University of Sussex recording radio when Derek Denton sent me a note saying he'd found a quiet retreat where he could write and I could pop in for a nibble. Well, I drove through divine countryside, noting so many polo fields and ponies and finally a posh drive that let me in. Derek Denton was in a cottage attached to a country house of splendid but tasteful appearance. I knocked on the door and Doris, the housekeeper, let me in. Derek was at his or someone's desk puzzling over a paragraph and greeted me in his thrillingly warm way. But I was thinking, if I was writing and the man from Porlock suddenly turned up, I'd be gnashing my teeth and saying, well, if you can't stay long, just stay for a bit. But But no, instead he took me on a tour of the grounds, which astonishingly led to a meadow and smart stairs that led down underground to a huge heated swimming pool, around which on every wall was the greatest collection of Mexican art you could possibly wish to see. "Where are we?" I asked, but to no avail. Later, Doris served dinner. Pheasant, of course, beautifully prepared, and wine, such wine. "Where are we?" I insisted before pudding. Derek Denton smiled and lifted the bottle he'd previously kept hidden. And there was just one word. It explained everything. The marvellous taste of the house, the art, and the funds provided for so much research at this university. It said simply, Rothschild. But so what? Well, Derek was there to write a book on the pinnacle of life, science and consciousness. When, in our last ever conversation, as we sipped our Bollinger, we were surrounded by books still open and papers still being written, just like Crick at the end. When Dick was 98, he was still working and working well. His wine was fine and surroundings often finer. But the priority was the science and using it for the benefit of humanity. And let's not forget, he was a splendid doctor still and knew more about my electrolytes and yours than many a specialist and his own. His art and his science came together as a universal empathy, understanding human engagement. It was emotional as well as intellectual. He was not deceived by the gloss or the frippery, which is why he ended his book, Pinnacle, with this reminder from Shakespeare. Our revels now are ended. These, our actors, as I foretold you, were all spirits and are melted into air. And like the baseless fabric of this vision, the cloud-capped towers, the gorgeous palaces, the solemn temples, the great globe itself, yea, all which it inherit, shall dissolve, and like this unsubstantial pageant faded, leave not a wreck behind. We are such stuff as dreams are made on, and our little life is rounded with a sleep. Derek Denton as Prospero. Vale, thank you.
Wow, what a speech, what a narration. Thank you so much, Dr. Robin Williams, for lending your great mind and deep thought to an oration that I think will go down in our memory books for sure. It's been a wonderful morning. I'm sure you're all looking forward to some bubbles and eats, which I think will be out in the foyer. I'd like to thank Trevor Kilpatrick, Angus Denton, and all of you who contributed to the morning for a wonderful time. And once again, to Dr. Robin Williams for delivering the most wonderful Derek Denton narration on arts and science. Thank you.